serving it. Ladies and gentlemen, it is a matter of immense pleasure to welcome you again to the National Seminar Water, Soil and Biodiversity Conservation, a shared responsibility for sustainable future, organized by the Department of Biosciences, School of Liberal Arts and Sciences, Mody University of Science and Technology. I present a very warm welcome again to the August gathering. President Ma'am, Dean Academics, Dean School of Liberal Arts and Sciences, all faculty members, students, and especially to all our esteemed guests. My name is Reem Fahim, and as your host for the day, I express my gratitude for your esteemed presence in this event. We shall formally recommence the seminar by playing the University Gul Geet. So I request the technical team to play the University Gul Geet for us.
यदि नैतिक आधार नहीं
Thank you. Today, we shall continue the spirit of the day before as we are joined by some exceptionally illustrious personalities who will shortly be addressing us. We are honored to have amidst us Dr. Om Prakash Ashwath, Principal Scientist, ICAR, National Research Center on Seed Spices, Ajmer. We feel immensely privileged to be joined by Dr. K.C. Soni, Professor, MGSU, Bikaner. We also have the honor of the presence of the respected Dr. Yogesh Joshi, Professor, University of Rajasthan. Thank you so much for sparing your valuable time for us today. Now, I invite Dean, School of Liberal Arts and Sciences, Professor Dr. Jitendra Binwal, to felicitate the respected Dr. Yogesh Joshi, sir. Thank you, sir. Biodiversity, the variety of life on Earth, is not merely a luxury but a fundamental necessity for the well being of our planet and all its inhabitants. Today, we are collected here again to be informed and to recognize the relevance as well as the necessity of water, soil, and biodiversity conservation. So, without further ado, let us move ahead with the seminar and begin the technical session number 3. This technical session is chaired by Dr. Navneet Joshi. Now, ladies and gentlemen, I am honored to invite the first speaker of the event, whose achievements in the field of agriculture, soil chemistry, and seed fertility and spices are remarkable. Dr. Om Prakash Ashwath, the Vice President of Soil Conservation Society of India, is the Editor-in-Chief of Global Journal of Biology, Agriculture and Health Sciences. Dr. Ashwath is also the editor of 4 national and international journals and newsletters and reviewer of more than 2 dozens of national and international journals. Dr. Ashwath is currently a principal scientist at ICAR National Research Center on Seed Spices, Ajmer. His contributions have been honored with numerous awards and medals. Ladies and gentlemen, please welcome Dr. Om Prakash Ashwath.
Honorable founder of this university, president, chairman, faculty, dean, students, ladies and gentlemen, good morning, namaskar, suprabhat. Before I have to start my deliberation, I would like to acknowledge the key person who involved in this program and invited me and honored me at this university. Dr. Navneet Joshi is a key person of this program, and he is in touch with me since the beginning, inception of this program, and he planned earlier workshop. Then I suggested him that I think it should be a national seminar. Then he made it like that. And Dr. Harish Kumar is also Dhingra. Dr. Harish Kumar Dhingra is also very much associated with this program. And very important and key person, Dr. Jiten Benbal sir. Dr. Sasiwala Singh, president, and the person who has founded this university, doctor, professor, or whatever the titles I can say, I think this will be very small for him. Sri R.P. Mody sir, founder of this university. I am really thankful all of you. You have invited me at this fora to express my views on my field of research, and I am really very much impressed with the students, faculty and staff of this university, their silence, their manner, and many more things. They welcomed me in that a very warm and a very loving heart. And so I'm really very much thankful and very much impressed, and all those things I will mention wherever I feel, whether it is in my newsletter or in that your book, I have mentioned everything. So I'm really, for your gesture, I'm really very much impressed, and I'm really thankful from my core of heart. So thank you very much to invite me at this program. And now I have to begin with this my deliberation. The topic chosen for this seminar is really very much concern for the present situation. That is very alarming. And the theme of this university is also quite related to this theme of seminar. And as far as my this deliberation, I have already worked on medicinal aromatic plant and presently working on spices. So what I found that these crops having the nature of these crops is quite different than the field crops. So I will not go on the about the general agriculture or something, because you have included everything, bioscience, agriculture science, and the technology, and very many more things we have accommodated here to make it very robust. And I am also in that field of agriculture. Yesterday, Dr. Balraj Singh, he was remained director for quite some time at my institution. So he has described about the general agriculture. Here I have directly focused on these crops. And why we are here, for that kind of seminar, because with the time we could not see the alarm of the nature. Whenever any situation goes wrong and we are moving somewhere from the right path, then definitely nature alarms us. If we are not looking into that, then we have to have its repairment by various way. So these crops are quite different than the field crops, and they are having wide adaptability. Ecologically, nothing is useless and nothing is very much useful. And like that, it is the nature. So these crops, where other crops fails, these can survive. So for the good thing, we are growing whatever the plants, whether it is ornamental, field crops or anything. But the situation, grave situations where we can not grow other crops, we can grow these crops and get the benefit, because these are very much adaptive to the stressful environment. And the secondary metabolites in these crops are synthesized only when the adverse situation is more, means stress is there. So we are a lot of seminars and symposia conducting on seminar on that stress, whether the human stress, animal stress or plant stress. But stress is also inevitable. If it is there, then definitely we can harvest the stress also. We can harness the stress also. And we can make the useless thing useful. As the government program is also there, that whatever we are doing, that we have to we have to produce waste to waste. So looking into that theme, I have chosen this topic because I am very much well acquainted with these crops, and few of them I have taken from the literature, and many of them I am well acquainted. So sir, our land degradation is going alarming, and this is directly linked to the climate change. Climate change is directly linked to the land degradation. These are our basic resources, and without that, the terrestrial ecosystem cannot be sustainable unless we will not look into that soil or the land. So the total arable land is only 3% of this earth, and you can see this earth pie. And some of that 9% land that is rocky, bad, hot, infertile, and these are the stresses. And this crops or the crops which are focused, we can utilize that 9% area without encroaching the main staple food crops. So we cannot stress the land and soil. What we can use judiciously in such a way so that we can supply the food for increasing and alarming population of the country. Sometimes we confused that what is land and what is soil. So here many of our young students are there, some professors are there, and that this field is also new. Even in the field of agriculture, many of we are not aware that what is the land and what is the soil. So this Venn diagram shows that land includes, the major is climate, relief, water, soil and vegetation. These are the 5 components of the land. So we cannot just synonym, or we cannot use the word land and soil very loosely. So soil includes that vegetation, and that is a part of that water also, and it is directly linked with that, but it is a part of land. Soil is a part of land, and soil is a now I have to again in this one I tell that what is soil. In general, it is phenomenal that around 25% of air, water, and 45% mineral, then organic matter, all those things it is composed of. And it is the soil and dirt, that's a quite different. Soil is a living body, and dirt is a non-living. It is just loose mineral matters and organic matter. So there are various type of land degradation, and that we have to manage. Otherwise, we may not fulfill the need of our food, whatever we are talking about our requirements in all respect. And these are various type of land degradation. And soil degradation, likewise, the soil degradation basically are the human-made and some of the natural. And we have divided, and this is not a part of this program, so I will not much highlight all those things. So degradation is going on various type of. If we talk about the extent of their degradation, so the worldwide, that 38% land degradation is there, but in our country, and total, that is arable land, and in arable land in country, our country is 44, sorry, 22.10 plus something. But if we talk about the total in total, then the soil degradation across the world is 52%, in India 44.7%. So we are still in a bit comfortable position if we compare with the world. What? India is a very diverse natural resources and natural these assets we do have, and we can harness them. Sometime I'm talking and arguing that we are very much focusing about the vertical biotechnology, but I would think that these terms is derived by me, that the horizontal biotechnology is very much needed to conserve over that various the diversity do we have. Actually, the 8% of the world diversity is in India because of wide this resources, and the land area is only 2.4%. So we are the rich heritage. Even some of the people, they are collected from NEH regions, many of our medicinal plants, some of that very important genotypes. European people and some Western people, they are having very monotonous environment. Their biotechnology is very much needed. Here we are in the name of breeding and many more research, somewhere we are losing in the name of discard. And those discard material is very much useful in the stressful environment. So that we have to conserve. So that's why biotechnology in India, that the amount we are spending on biotechnology, we are not getting that much of output from the biotechnology. That is one of the reasons. When the things are available with simple means, why we should go very exhaustive or expensive means? So this is the land degradation across the world that we can perceive that where degradation is very highest. So see, our country is also is alarming situation. If we look into that color combinations, based on that they have depicted, and like soil degradation also, we are also in the very alarming situation. So how do we control this land degradation? By using our huge biodiversity. We have different crops in that food and staple we have lost because of the high yielding and high fertilizer responsive, and because of that reason we are not having those material which could survive, and which was that was having a very good inside combination of the nutrients. So, and that's why we are feeling malnourished, because of we are getting too much of that food. Even then we are feeling malnourished. We are gray hair color because of that zinc that is not sufficient, or it is there, though we are telling that biofortified, but still it is having a deficiency because the bioavailability is not there. Whatever we are consuming, it should be absorbed by our body, then only it can translate into its functioning. So how do we manage? There are various management system on this degradation, and without that considering this mechanical and all those things, we can control through our crops, whatever we are food and taking from them. And for that we can use our lower plant like that after this there will be a deliberation on that line also, bryophyta, thallophyta, pteridophyta, like that. Higher plants, spices, medicine, aromatic plant, and other crops of the field crops, food crops, staple crops. So I have to started that land degradation from the eroded land. How that we can control the erosion by using these crops also. So we have the dual benefit. One side we can control the erosion, and other side we can get something from out of that degradation or that problematic area. This is a photograph of that Cherrapunji. Here you can see the Pandanus odoratissimus, that is the kevada. In Hindi it is called as kevada, that big plant you could see. It is on the cliff of the outcrop of the these rocks, it is existing. You can see from the photograph. And the grasses you could see, this is the vetiver. That can be used as an aromatic plant, and the roots we are extracting the oil, and that is very useful. And so we can use this crop where nothing we can get some earnings, we can get something good, we can get the perfume from that. And odoratissimus is the edible oil, and generally our big programs we are using this oil of this kevada, Pandanus odoratissimus. And these are the plants we have listed for that eroded land. These eroded means water eroded land. And then Anathem soa, that is called as soa in Hindi, and Osimum, that is tulsi, Semopogon species, these are aromatic grasses, Eucalyptus, Lavandula officinus, Rosa domestina, like that, Mokuna, this medicine plant, Barleria parentis, Bajradantri, Agave, that we know this is ornamental as well as the medicine plant also, like that Plamago jalanica, this is Chitrak. So these all we can take under water eroded soil, and the grasses can help to control that seat erosion and rill erosion. This is a dill crop. You could see many of our students, those who are from the city west, and they are not heavy in testing for them. This is the Osimum tulsi, and in the inset it is a tulsi that is given in that inset, and this is a Ram tulsi, Osium sanctum and Osimum, other species also. The Mokna, for instance, is a leguminous plant, is very important. This can cover the crop so that the by rain beating, whatever the loss is there, that we can control. So it is mutual benefit. And the left side, this one, this is Vajradanti that we are using in that our teeth, this one, mouthwash. This is the agave, various type of agave, our toothpaste and all those that we are getting. This is the Vajranti, Barley apparatus. Chitrak, it is a very important medicinal plant, and there are a lot of stories about it, that that even a wire can melt down if we touch with this stick of this Chitrak, that is the Plamago jalanica. Likewise, Amla, Amblica officinals, everybody know about it, nothing to say about it. That is a very important for gully erosion. It can control the gully erosion. Gully erosion, the erosion is in the water erosion. First level of erosion is the seat erosion, then is the rill erosion. Small gullies are there, then a big gullies are there, then it is called as in that it is a very useful. Even in dry conditions, this is the photograph of Bikaner. There we can get that Eagle marmalus whale. We all about it is important medicinal property, and how it is very useful for that stomach problems. That we can take in that soils. Chlorophytum borebilianum, Safed musli. There is one of the important medicinal plant in the travel area. Travel people, they are using in place of your whatever the you are getting that tonic. It is an tonic. After delivery, they are taking its milk pudding, and they are so healthy. I have surveyed in the MP, and this is a very important medicinal plant. You can find this roots of in the market. It is available. This can be grown even a fist of soil. It can comes up in the rocks. Crevices are there. There it can comes up that Chlorophytum borebilianum. This is I am giving the photograph because this I have taken at that Nohakalikai. There one lady was committed suicide, in the name of that place is known as Nohakalai, that the 0 kilometer. This photograph I have taken. Many of that photographs, they are from my own I have taken, because I am across the country. I am moving and delivering all those things wherever it is required. And if we talk about the erosion, then it is a wind erosion and the water erosion. And the sum of that term that I have coined, that is nutritionally eroded, because whatever the nutrient do we have already mined them from the soil, and that is exhausted. So how do we use those soils where the other food crops we cannot take? Because we are telling that this is a poor supplying capacity, or they are the deficient soil of the nutrient. There we can also grow some crops. These crops are having capability to draw the nutrient or to mobilize the nutrient, or some of the crop they are having capability to economize the nutrient by translocating and retranslocating. And one of my students, that abstract is also at the last, you have listed there. I have given that work, that how plant economize. Because when the adverse situation, how to cope up that adversity, plant have mechanism. Initially it accumulates, and as soon as it is getting the congenial environment, or at the time of its flowering, seed setting, means for that biological requirement, so that it can continue its cycle, it is accumulating and then transferring to the its genetical part, particularly flower, and so that it can set the seed. And by that way, it can, whatever the nutrient accumulated, it transferred to the seed, so that its generation could continue, its life cycle could continue. So that kind of mechanism, so that we have to identify all those things, so that we can utilize our very squeezed or the eroded nutrient situations. So some of them are having capability to use some microbes, and some of their requirement, they have minimized our requirement. So this is the requirement of the nutrient of different crop. And the coriander we have listed here, it is in the southern area. Their requirement is quite low because their output is also low. But in the northern India and middle India, we are having high yield potential. Their requirement is quite high. So we cannot count them at a very low requirement. Chlorophytum borealis I have listed in the last, and that is a very important crop that I told that with very small amount of soil it can comes up very well. So those situations we can utilize. And some of the crops, they are having colonization with the microbes, with this vascular and arborescent mycorrhiza. And they are having mutualism or symbiosis, we could say that, by taking that carbohydrates from the plant and supplying the nutrient by a big mycelium spread in the soil. So drawing the water and nutrient from the soil and providing the plant, and they are getting carbohydrate from them for their own life cycle. So that we have listed some of the work we have already done in that line, some of the crops, and some of I have taken from the literature. So they are having good affinity with these microbes. So by this mutualism, they can survive, and they can win-win situation by giving and taking from each other. And vetiveria, like that metha, peperata, that metha, this peppermint, and ये जो हमारा रहता है ना पुदीना, इसमें सबसे highest affinity है, because its requirement is quite high. So the plant is also seeking ways and means that how could it fulfill the requirement. So that kind of they develop the mechanism. This is Gloriosa superba, very poor soil. The colchicine we are extracting, we are using our laboratory. That is comes from this. It is a very good crop, but it is a very unfortunate that it is in verge of extinction, because our requirement, and it is having one of the important problem that when any insect visiting for this pollination, its nectar is also having that poison. That's generally we are doing this colchicine for this staining the cells, or stopping that meiosis and mitosis. So in that condition, here is so whatever the insect visiting, it dies after taking nectar. So it cannot transfer the pollens to the one flower to other. That is one of the problem. Like that. This is for the compacted soil. Compacted soil, some of the crop size vary. Where other, even you cannot penetrate a khurpi in that soil. spade, you cannot cut that soil. You can grow under those situations these crops. These crops are the medicine Sena, Cassia angustifolia, that is also a Sanai, Sonamukhi. And Tivulus strestis, it is a gonkuru, chota gonkuru. Pedalium murex, there is a bada gonkuru. That we can grow in the desert. Even this photographs from the Sam dune in that Jaisalmer. And there we can grow. When nothing is there, we can get these crops under those situations. And by that way, we are stabilizing the sand dunes. By wind erosion, we can control by these crops. And some of these crops I have listed here, that Amimagus, Pampelana, anisum, for hot and dry area conditions, including that compaction, Phoniculum vulgare, trigonal, that some of these spices are there. And that Ferula asafoetida, that is hing that we are using, that is also ambellifrant. Artemisia maritime, Semopogan, Gugul, it is hardest for the dry and arid areas. Some medicinal aromatic plants, that isabgol, Acaranthus, Ocimum, Acaranthus aspera, Lavandula officinals, Rosa domescena, Aloe barbadensis. Yesterday we were talking about that aloe. And Calotropis. Calotropis, as soon as the that temperature shoots up in the desert, its growth becomes luxurious. It is a that heat and stress-loving plant. I will show some photographs like that other plants also. This is Senagin Cassia mustifolia. This is a very dry area. And this Halotropium, this is a ornamental plant as well as a medicinal plant, and that was growing in that highly saline soil or high temperature. When the temperature shoots up in May and June, its flowers are it is blooming like anything. For the moisture stress, Plantovobata is one of the important crop, particularly for Rajasthan and North Gujarat. But where there is a late winter rainfall, it is not good for that areas, because it crop destroy. Its gel become like this thing, that crop is failure because of that reason. This is Ferula asafoetida, that is a hing plant. I think we all are taking, and probably you are not aware that how that hing extracted from the plant. It is a latex, just like this one, Papaverum office, opium poppy, sorry. In the same way we are collecting. This is the Amimagus, this one, Ajwain, dry and poor moisture supplying, says Salvia officinales, Artemisia maritime, medicine plant. This is Gugul, that we are taping the its gum, that is very useful for heart diseases. This is inside of that same photograph. This is in that plant I have collected from Rajasthan and Kutch region, and I planted in that Anand, Gujarat, when I was working there. So this is my own collection. This is Calotropis that I was telling. Calotropis, this is one of the medicinal plant for not only for human being, for the soil, and some for insect and pest management. This is one of the important crop, and it is going lust green in May or June, when nothing vegetation is there. This is the second photograph from the. This is the again these crop having multi-facets. Somewhere moisture stress, or somewhere temperature high, and somewhere that soil compaction. Likewise, somewhere this common. This I have taken from the same that sand dunes. This plant is, you can see, this is a rotundus, and this is also a very important plant. When nothing vegetation you could see, it is persisting, and it is having wide adaptability from submerged condition to very dry area. And some of the saline alkali soil and sodic soils, these are very big problem of endogangetic plant, particularly from Karnal to beyond the Kanpur. That stretch is going, and here most of the soil are saline and sodic. In Rajasthan also, underground water, most of the soil are saline and sodic, and underground water is also. So there we could take some aromatic plant where ESP, axial sodium percent, is more than 50%, or the EC, electrical conductivity or electrolyte concentration in soil is around 10, 12. We can take these some Pomarosa, lemongrass, vetiver, Jamarosa, citronella. Salvadora persica, it is one of the important medicinal plant where nothing, even miles together, you cannot find a single plant of vegetation. It can grow in that soil where the EC is around 60 or more than 60, where you can see the surface of the soil is completely white, and nothing vegetation is there. The crust of that this one salt is in inches. There you could find this plant, Salvadora persica, and popularly known as Pilua, and in Gujarati Pilodi. Even in the saline sodic among the seed spices, this cumin is the highest salinity tolerant. It can tolerate ECE around more than 30, and if EC is 1:2.5 of water, that is more than 13 or 14. It can tolerate. It comes up very well. Even the salinity is desirable for this crop. That I have established, and the first time I have reported all those things. That the seed filling is quite better when there is a slightly water is saline, but it is sensitive to sodicity. So if the EC is around 4 of the irrigation water of the soil, the filling of seed is quite high. In the same gunny bag, you can fill more than around 25% extra seed weight than the actual one. These are some of the crops seed spice we have taken at Hisar, that the regional station of Karnal Institute, that is a CSSRI, Center Soil Salinity Research Institute. There I have done with collaborative work with those people. Likewise, Pomarosa I have already worked. This is also one of the important, but this is a very hardy, but at its juvenile stage, it is very much sensitive to the sodicity. So liming sclerosis is persisting where the lime is quite high in the soil and pH is also high. Lemongrass, that is also very hardy for salinity and sodicity. Likewise, vetiver, it is submerged to dry area. It's having wide adaptability for salinity, and you could search the literature. There are a lot of international conference, particularly on this single plant, and huge literature is there. It is also act as a biofilter in the soil. It purifies the water bodies. Some aromatic crops, they are sensitive to sodicity, so that photograph you could see. So that we have not take like Jamarosa, the right side. Eucalyptus, whatever the hybrids we are developing, the original natural species are there, they are more tolerant than the hybrids we have developed. That figure is showing of that kind of. This is eucalyptus. There we are using that. This Vicks vapor of, you can find the same smell. It's the extract of that one. So it is a very important aromatic and medicinal plant both. Sodicity, some of the sodicity in terms of pH, that Pomarosa, lemongrass, they are having capability to tolerate the pH 9.5 or 10 like that, and that is very alarming situation for other field crops. This is the fennel. Fennel is also having strength for the sodicity. Even its requirements also there. When we are giving sodic soil, it comes up very well. It mines the salt, so it can improve the saline sodic soil also, and is having very good capability to withstand in the compacted soil. Even soil volcanicity goes beyond the 2.8, it can survive in that also. Even along the roadside, like the tuberous status and Pedalium murex, it can also grow along the side of the road. Even the paved road, in the small crevices, it can comes up because it's having capability to penetrate that hard soil. So compacted hard soil, it is very good for that. Like Ambretti, that is also medicine plant. Likewise, this Lavandula officinals, lavender, that is we are using as aromatic and perfumes. In that we are getting the odor of these plants. poor quality water. That is very much concern to Rajasthan, that around more than 80% of underground water either saline or sodic. And under those situations, we can grow many spices, medicinal aromatic plants. Polluted and contaminated soil. I have listed lot number of these plants. There we can grow them, polluted and contaminated soil. And they can purify that water, and they are resistant in many of the aromatic plants we can take, because we have to extract the aroma for our commercial purpose, and whatever the left behind we can dump in a small place so that it can be used as a bioaccumulator. Some of that irrigation water that we have experimented for various crops, and their degree of tolerance it is given in that. And in that saline sodic water, this vetiver is also coming up, that salicornia. It comes up very well in that water inundation from the seawater. In that also it comes up. It is a very good medicinal plant too. This of the celery, it is the highest tolerant of the salinity after the dill. And that experiment we have conducted that kernel, and the person is with me. He is Dr. Yadav, R.K. Yadav. He is presently a director of that institute. This is Pudina. Acid soils, not only these soils, these acid soils are also, these crops are very adapted. And these crops, coriander is called as a bowl of that coriander, this seed spices at Rajasthan and Gujarat. And coriander is known for that Harodhi region, but it is extinct from that place. And we are getting the yield around 16 quintal, average yield, or means highest yield we are getting 15-16 quintal in that area. But when we have taken this in the acid soil in the northeastern hill regions and eastern plateau hill regions, there we found that it is giving more than 30 quintal per hectare yield. So it is a acid loving crop. So slightly acidic soil is quite better. In anise, anise is one of the important seed spice, as well as in the high altitudes. This crop comes up in acid soil quite well than the this situation. Their quality and quantity both improves, and we can this also anise is also used as a tea, and its decoction can increase the capability of lungs. And sometime we are finding that some doctors, they are giving some these pouches, and for that steam purpose, and it improves the lungs capability. So those who are climbing high hills, they are using the tea of this plant. And this is moringa that we know very well. Cinchona, that is used in that quinine extraction. Like syphilis and coptis, fennel, that also comes up. Dill, nigella, with a slightly acidic soils. This is anise that I am talking about, that for lungs capacity, Pimerella anisum. This is the nigella. It is one of the important medicinal as well as spices, generally be used for the pickle purpose. And these experiment I have conducted at eastern plateau hill regions in Ranchi and other that northeastern hill regions. My friends, Dr. B. Keja is there. He was the co-PI of that center. And moringa, that we all know, huge stress everywhere we are giving for this plant. It is having wide adaptability from acid to alkaline soil and moisture state to waterlogging area. And it is a very good plant for all those conditions. And its drumsticks we are using in the foods. And southern India, we are using huge amount of this plant. Even in Gujarat, you could find huge and very big farms are there. And we are getting that drumsticks from that. This is cinchona. This is syphilis, coptis. Flood and waterlogged area. There we could, I have already mentioned some of the plants like vetiver, pandanus, elateria, cardamom, that is elaichi, Acorus calamus, medicine plant, metha, this is pudina, and this peppermint we are using, that is only extract or the essential oil of that plant, and we are, when we making it condense, then it is a peppermint, this dry form. like Ziziphus aromaticum. So this is a cardamom, electric cardamom. This is a cardamom plant. I think many of we are not aware that what kind of this cardamom plant is there. Choti ilaichi. And that big one is in that north eastern hill regions, because it is having its specific quality, and that pollination is required for the bumblebee, and that is available in the northern eastern states. So that big ilaichi is not available in southern India. Bacopa monareri, that is the Brahmi. This is a Thrissur Brahmi, you could see. In southern India it is found, and in the northern India, Centella asiatica. That is Haridwar Brahmi. It is also called as. It is a very memory enhancer, very important medicine plant. That can come along the side of water bodies and the drainage channels or the irrigation channels. In that water bodies, Acorus calamus, that is called as sweet flag, is one of the important plants, comes up very well in even submerged conditions. Salicornia, again it is coming for the salinity also, sodicity also, then it is again for the submerged conditions. This pandanus, it is lagoons. You could find along the side of seawater inundation is there, or particularly in that Sundarban area, you could find that this pandanus odoratismus is there, Kevada. Metal contaminated soil. In nutshell, I would say, whatever the aromatic plants we can take, metal contaminated soil or the polluted soil, because we are not using its part for the direct edible purpose. We are extracting perfume or the essential oil from that. So in that area we can use those soils by using this biodegradable. Besides these crops, their varieties are also having different degree of tolerance for different these adverse conditions I have mentioned, like erosion, like that chemically degraded soil, acid, alkali, like that and that. So their varieties are also having different degree of tolerance. So we can use them also. So in that conclusion, I would say that I have covered all those aspects. These are the very major. If we can control the erosion and land degradation, we can control the climate change. And I have also graduated from a Nobel laureate, Professor Zanlal. He is a father of carbon sequestration and climate change, and under his leadership, I have also graduated from the Ohio State University, Columbus, and he always emphasizing that for the how do we control the land degradation, because the ephemeral zone of the globe, that is highly vulnerable. Though our Indian condition is not that much of in that grave situation that we have to be very serious, but because we are having huge biodiversity, and we are having the shifting paradigms when the temperature will rise, so we will not that much affected except the coastal area. But these European people and these some Western people, they are located in the high altitude area. They are very much vulnerable. Particularly ephemeral zones means the zone in between the ice encapted and the non-ice encapted area. In those areas, that is called as ephemeral zone. Though all those things we have created in this country, our sages, our priests, our intellectuals, they have already preached that how we are facing this problem of this degradation, and so many environment, health problems are there. Why? Because we have not followed our moral. We are overusing, misusing our resources. Because of that reason, we are facing all those problems. Why I have put this James Gustav's this photograph and this thing. Now these people, those who have whole of their life, they have advocated that how to conserve the biodiversity, how to control the climate change, how do we control the land degradation by various means, this technology and that technology. But ultimately now they are telling that without the moral or without the looking into that its value, we cannot conserve, we cannot change this, we cannot stop that change we are going in that climate change or degradation. As we know, it originated from our country that we are worshipping the nature. Indraho Devata, Rudraho Devata. Though there are so many problems created in the name of like Indra, even then we are worshipping them. Even the Nag, one day we are worshipping on the Nag Panchami, and another we are trying to kill, because when there is a situation when it is harmful to us, then we are take care for that also. So they are now, they are aware about all those things. Then they are propagating, we are implementing. So we should, we were the leader, so we should remain the leader. That's what I have quoted. We should not go to quote with them. But before quoting them, I would say that we have to quote our Mahavir Swami, our that Gautam Buddha. They told that how do we attain that salvation? By abstaining from all the pleasures. We are very much concerned with the physical pleasures, and that the Western people, they are advocating many of the things, though some of the things they are advocating, that is very good. But the things which we have already know, and we have that heritage, that we have to think, and we have to respect our forefathers, what they were they having. And sometime I used to quote that the way we are progressing for that high technology, this and that, but other way around, morally we are somewhere going down. Yesterday, there are a lot of technology, and these things we were hard, and every platform we are listening. But what is the level of its use, that we have to devise? Because whatever the technology we are developing, this is useful, this is unique, this is everywhere. No, every technology have its own limitation. We have to define, like some very good or high impact factor journals. They are asking what are the drawbacks of your research, and that bottom of line they are mentioning. These are the limitation of my research article, that we have to mention. So that person cannot deharvest its demerits. One of the example I would like to give, that one of the our crop, it is a very good crop, and one of our NGO, he has given to some farmers that it's a very good and it is giving very good yield, and that crop comes up in a medium fertile soil, and they have given in that dhoras of that dhors of the Rajasthan, means sand dunes, and that crops was failure. So we have to decide. See, there is no single theory is foolproof. Every theory is having in its own limitation. Like one thing we used to say, every theory fails. The one theory is always persists, that is death. But no, that is also our Lord Krishna says, न जायते म्रियते वा कदाचिन नाय भूत्वा भविता वान भूया अजो नित्यो शाश्वतो एवं पुराणो हन्यते हन्यमाने शरीरे. He made the correction. No, we are not dying, neither we are reborn. Our Atma, our soul is getting new body, just like clothes. So he has also put the clause on that. So we have to, my message is to my peer workers, my students, you be honest for your, if you are studying, you study honestly. Whatever you do, you do honestly. When I have attending the convocation at Ohio State University, Columbus, and that time, Vice Chancellor of that university, he emphasized to the student that it is a take-home message for you, that whatever you did, it was your training. When you are moving out, your real examination will start. If you will prove yourself in your workplace, it is your real examination, you will pass or fail. That will depend on by your action. So it is not only for that the knowledge we have acquired, and now we are knowledges, because sometimes we are feeling that I have attained the position of this and this, and we are thinking about this, but we forget what is our responsibility, what I have to do. Sometimes we are showing the power in the form of that we can do this one, means for the punishment, but no, we have to see, this is my message to my elders or the senior positions, how we can facilitate the people for that position, then it is my power, otherwise not. So thank you very much, and one more thing I would like to say, that we are also organizing one national conference at ICR National Research Center on Seed Spices, Ajmer, and that is on 13, 14 and 15. So you are also cordially invited, one and all, if you have any kind of your paper presentation, and we will accommodate, and I would like to see you, some of them, from that place. So you are cordially invited for my this conference also. Thank you very much, one and all, and I'm really quite happy. I'm thankful that the way you have listened, I will never really forget this whole institution and university. Thank you very much.
So, from behalf of Mody University family, I thank Professor Ashwath, Principal Scientist, working at NRCSS Ajmer.
Okay, thank you very much. But before asking question, I would say about me that a scientist is whatever question you can ask, you're welcome. But a scientist cannot answer every question. A professor can, because a scientist, if he will not concentrate and he will not serve itself, it cannot be a scientist. Otherwise, it cannot be penetrated deep down. एक जो कील होती है ना, उसको भी हम साफ करते हैं उसका, so that it can. हाँ, please. अब माइक देंगे क्या? अच्छा, मैं सॉरी अंग्रेजी में प्रेजेंट किया. Any हिंदी, इंग्लिश, whatever the way you want to discuss, I will discuss now. और अगर किसी को समझ में नहीं आया है अंग्रेजी वाला, जैसे कल बोल रहे थे कि ये सब लोग अंग्रेजी में बोल रहे हैं, तो I can speak in हिंदी also. Even Gujarati. Because 10 years I served there, so I learned Gujarati. And Gujarati is nothing but it is some mixture of Marwadi and Marathi, and that is a Brij Bhasha, basically, Gujarati. Sir, please. That's all.
Topic all, but there are certain. I was just wondering, so I just want to put into words.
Yes, please.
So first is, as far as I know, most of the spices, apart from lavender, cumin, they were harvested in a larger amount. But why is it most of the other things remains as a wild herb? That is the fun. I'm just wondering why most of the things remain as a wild herb. Let's say I'm from the hill area. So there are small herbs that is very good for the medicinal purposes, but it was not cultivated in a larger amount. Okay, sir, can I continue my question? Otherwise, I will forget.
Okay.
Yeah, another one is, let's say, so in between, sir mentioned about the one plant, I couldn't recall the name of it, which helped as a to repel the insects.
Mm-hmm. That's Palmarosa lemongrass. Lemongrass is one of the Java citronella.
Okay, sir.
It is used in that odomas.
No, sir. I mean, is there any more which can help? Is there any plantation herbs which can help? Let's say if we plant in between the field of the wheat or the rice, which can repel the insects.
Yes.
That one. And the third one, is there any specific spices that work as a nitrogen fixation plant, if it is there?
Yes. Seed spice. Yes, I will explain. But your question is really very pertinent. First question is very pertinent, very important. People really rarely interact on that. We are doing very hi-fi research, genomics, proteomics, this and that, and me also. I'm not out of the system, including me. What we are not aware about what is the autoecology and synecology of the crop. That's why you could find in the nature, but when you will bring in the cultivation, because what we think, it is good, because it is a good for a particular crop, not for all. Whatever we are devise the congenial conditions or good condition for wheat, barley, sugarcane, that cannot be congenial from the other. So we are thinking this is the good. It is not good for all. Likewise, our medical science, we are taking some of the benefit. So if we are not the self doctor, then nobody can save you from the doctors. So this is the situation. The species, their requirement is quite different, and we are thinking this is the good, this is the good. No, it is not always. Some good things are bad for other plants, and that is the diversity. That is the biodiversity. And it is a huge number of group of the plant for your second question. Like nematodes, some of the plants like hajara, that we call that is a repellent for the nematodes. Likewise, it is the host specific plants. Some plants, they are repellent to particular insect species, and some of them for the other one. So we have to choose that what kind of infestation, what kind of insects infestation they are, and according to them we have to plant them. That's a big area. I cannot answer in a single or this question answer session. It's a big area. So that we have to identify. Even if we place them or plant them in a right place, then very few diseases will come, because these plants having inherent capability to withstand or to cope up or to save from these. See that injury, when there is a fungus attack, so there is a lesion, and PCD, programmed cell death, it is a cope up mechanism. It will die there, the plant will die the cells, so that this infection cannot spread further. Only they can lose the limb. Sometimes doctor says, you are having a very big infection, now I have to cut down your limbs. Then otherwise it will go in whole orthopedy and you will die. So like that, plants are also having good mechanism. So if we are going in that right place, if we are giving proper nutrition, then insect pest and diseases are very, very small, very, very less. Nature having its own resilience power. Please.
Namaskar, sir. My name is Dr. Pramod Kumar. Just I want to know, sir, the production of cumin and coriander is limited to Gujarat and Rajasthan. So is there any research going on to produce, to develop any variety of these spices so that they can develop in different states, having different soil conditions?
Very good, very good. बहुत अच्छा आपने प्रश्न किया है. Actually, 15 years, more than 15 years, I am at that institution. We are very much concerned about our own promotion, rather the crop and this thing. So that is one of the reason. That is officially, and at the grassroots level, I would say that cumin is highly susceptible for blight. So where there is a high moisture or the humidity or rainfall during winter season, it will wipe out. Though it can germinate many places, but it cannot go up to the seed setting. So that's why it is limited to the area where snowfall is also less, like Isabgol, Plantago Evata. These are limited in that Rajasthan and North Gujarat. This is so that we kind of we should have some variety which can comes up in that condition also, then it can spread that area. And it is the economics, demand and supply, because its demand is quite high than the supply. That's why its price is quite high. It went of 700 kg. Yesterday Dr. Balraj Singh told. So if you will produce more, like fenugreek, few years back, but we have seen that the production increased and the price went down half. Then what is the, how we can get the doubling farmer seed income that has governed by the government? And all the time I was say, no, I cannot follow this one. We can double the yield, but to double the income, that is not our hand. That is depend on the market.
But sir, one thing, in the previous year, the cost of cumin per kg was near about 450 rupees per kg. That was. But now the cost of cumin powder, cumin seed per kg is near about 900.
Last year.
Yes, sir.
Last
How
year.
This difference, sir, created.
It is saying in the farmers, within the 3 years, you can get a good crop only once. So there is a huge crop failure last year because of this reason. So supply was poor and demand was quite high. And that's why price goes up. Until unless you will purchase, they will sell like anything. Like your onion, people are in the line, in the queue for the purchase of onion. Because sometimes that market people, they are creating the crisis, artificial crisis. नहीं है भाई, प्याज नहीं है, अब क्या है, कहाँ करें? लोग लाइन में लग गए और वो खरीदने के लिए, जबकि क्राइसिस थी नहीं, उसके बाद शुरू हो गई, आना शुरू हो गया. जैसे कि बहुत सारी ऐसी चीज़ें हैं कि हम इंपोर्ट कर रहे हैं जिसको इंपोर्ट करने की ज़रूरत ही नहीं है. हम 10% एक्सपोर्ट कर रहे हैं, दैट इज मीट द 50% डिमांड ऑफ द वर्ल्ड ऑफ द क्यूमिन। तो हम लोग बहुत स्पाइस खाते भी हैं। और व्हेन मैं जब बाहर कंट्री में था तो मेरे से कहा कि सैनिटाइज़र यूज़ करो, ये करो। हमने कहा हम लोग इतना स्पाइस खाते हैं, जनरली हम लोग इतना इफेक्ट नहीं होगा, इसीलिए कोरोना में हमारे यहाँ इतना ज़्यादा प्रभाव नहीं पड़ा जितना कि यूरोपियन कंट्रीज़ में रहा। इतना हमारा एसेंशियल ऑयल है, और एसेंशियल आप सभी जानते हैं, सारे लोग जानते हैं कि जर्मिसाइडल प्रॉपर्टी है इसकी, और बहुत अच्छा हमारा काम करता है। हमारे फोर फादर्स ने, जैसे नाइजेला को लोग बोलते हैं कलौजी, कलौजी है, आपको हम अचार में डालने की टेस्ट उसका अच्छा नहीं लगता, लेकिन हमने डेवलप किया टेस्ट। हमारे फोर फादर्स ने उनकी वैल्यू जाना, जो हम बोलते थे ना, हमारे वो दादी माँ की जो किचन से ट्रीटमेंट करते थे, तो वो इसलिए हमने रखा, किसी न किसी फॉर्म में हमने लेना शुरू किया। आप अजवाइन की पूड़ियाँ बना लीजिए, आप 2 दिन तक 3 दिन तक कुछ ख़राब नहीं होगा उसका, वो डाल के। तो बहुत कुछ है हमारे पास में, आज लोग हमसे बाहर वाले सीख रहे हैं, लेकिन हम उसे भुला बैठे हैं। अगर हम ये बात कहें तो हम पॉलिटिशियन हो जाते हैं। बहुत सारी चीज़ें हवा हवा में चल रही हैं, अच्छा नहीं लगता बोलना, बिकॉज आई एम ए पार्ट एंड पार्सल ऑफ दैट सिस्टम। तो जब तक हमारा मॉरल नहीं होगा, हम सही चीज़ को सही नहीं बताएंगे ना, तब तक कुछ नहीं होगा। कई बार किताबों में वन ऑफ माई रिलेटिव इज इन दैट रोड कॉन्ट्रैक्टर, उन्होंने एक रोड का कॉन्ट्रैक्ट लिया और वो कहा कि इसमें पैच वर्क होना है, और जब वो साइट पे गए तो देखा रोड है ही नहीं, इट वॉज नेवर देयर, और पैच वर्क के लिए उन्होंने कॉन्ट्रैक्ट लिया। तो होता है, चलता है ऐसे। तो जब तक ये चीज़ नहीं होगी, हम ठीक चीज़ को ठीक से नहीं परसीव करेंगे, तो नहीं, हमारे पास बायोडाइवर्सिटी है, इतने वाइल्ड स्पीसीज हैं कि हम सिंपल क्रॉस करेंगे तो ये फ्रूटफुल है। यस, यूरोपियन लोगों के लिए इनके साथ बहुत बड़ी समस्या है, इतना मोनोटस एनवायरनमेंट है, मोनोटस स्पीसीज हैं, अगर वो वर्टिकल बायोटेक्नोलॉजी नहीं करेंगे, वेरिएबिलिटी क्रिएट नहीं करेंगे, तो वो कुछ नहीं कर सकते। इसलिए उन्होंने पूरी एनर्जी लगा रखी है उसमें। हमारी तो जो एग्जिस्टिंग बायोडाइवर्सिटी, उसको अगर हम हार्वेस्ट कर लेना, इट इज़ मोर देन एनफ। एंड आई कैन चैलेंज एट दिस प्लेटफॉर्म, यू कैन रिकॉर्ड इट। हमें कोई क्लाइमेट चेंज एंड डिग्रेडेशन से बहुत ज़्यादा फर्क नहीं पड़ने वाला, अगर हम थोड़ा सा ध्यान देंगे तो। बस थोड़ा सा ध्यान देने की ज़रूरत है। इसके पास हमारे पास बायोडाइवर्सिटी है। मैं खुद आई एम ए सॉइल साइंटिस्ट, बट मैंने चैलेंज किया डॉक्टर चुंडावत साहब, जो वाइस चांसलर बहुत लंबे समय तक रहे हैं गुजरात में और राजस्थान में भी, तो उन्होंने कहा कि आप मेरे को ये जो कोरिएंडर की वैरायटी आपकी है ना ACR1, मैं एक वैरायटी से 8 वैरायटी निकाल दूँगा। तो वो उनको थोड़ा सा बुरा लग गया कि इट इज़ ए चैलेंज फॉर द ब्रीडर्स। सब ये ब्रीडर्स के लिए चैलेंज की बात नहीं है। 5 की मैं गारंटी देता हूँ कि मैन आई एम गिविंग द स्ट्रेस, बिकॉज आई एम वर्किंग सिंस फ्रॉम बिगिनिंग ऑन दैट स्ट्रेसेस। सो व्हेन आई एम गिविंग द स्ट्रेसेस, इसकी असलियत खुल जाती है, तो उसमें से नए नए प्लांट्स निकलने लगते हैं, वैसे दिखने में सिमिलर दिखते हैं। तो बहुत कुछ है हमारे पास, इतना कुछ है हमारे पास, हेरिटेज है कि हम जहाँ जंगल में मंगल कर सकते हैं। वी कैन टर्न डेजर्ट इनटू द ओएसिस, बट वेरी अनफॉर्चुनेट सम पॉलिटिकल प्रॉब्लम्स आर देयर। अब पालीवाल साहब का उदाहरण है ही, यहाँ पर कि माइंड सॉइल में, और उन्होंने कैसा हरा भरा बना दिया। बहुत कुछ है हमारे पास जो है एग्जिस्टिंग, और उसको हम समझ लेना, इट इज़ मोर देन एनफ। और फिर सब लोग इतना वो प्रॉब्लम्स को पोज करते हैं। हम अगर चाहें तो हमारे पास जो सिचुएशन है, हमें बहुत घबराने की आवश्यकता नहीं है। जैसे डॉक्टर डरा देता, अरे अब आयो, बहुत लेट हो गए, अरे आज नहीं आते ना तो आपको ऐसा हो जाता, कल नहीं आते तो ऐसा हो जाता, आदमी हाँ घबरा गया, एडमिट करो इनको, ऐसा। और वो इसलिए कह रहे हैं उनका एक बेड खाली पड़ा है, उनका एक वेंटिलेटर खाली पड़ा है। इस तरह से हम लोग, हम किसी मेडिकल डॉक्टर से कम थोड़ी हैं ब्लफिंग में। ये मैं अपनी बुराई किसे कह रहा हूँ, ये इसकी ज़रूरत है। हालांकि प्लेटफॉर्म इस तरह करने के लिए नहीं है, बट वी हैव टू गिव द मोर स्ट्रेस ऑन द मॉरल, एंड दिस इज द एजुकेशन इंस्टीट्यूशन, देयर वी हैव टू थिंक। हम अभी से इनके मन में डाल देंगे साहब इम्पैक्ट फैक्टर में छापो, अरे जाने दो ना, क्या मतलब है। अभी जो लोग दावे ठोंक रहे थे, मैंने बोला किसी टेक्नोलॉजी के लिए मेरा नाम इसमें से हटा दो, हटवा दिया, क्योंकि इट इज़ फेक। और पब्लिकेशन कुछ भी होता है, मैं अपनी बुराई कर रहा हूँ, मेरे MSc के 3 पेपर हैं नेशनल जर्नल में, और तीनों फेक हैं। मैंने जब अपने गाइड से बोला कि नहीं सर ये नहीं, रिजल्ट तो ऐसे नहीं ऐसे, डू यू थिंक आई एम योर गाइड और यू आर माई गाइड, यू विल नॉट गेट द डिग्री सर प्लीज़। जी
acidity or removing water, है ना, So what variety you have developed using the stress condition?
For me, this
For-
variety means for my seed spices.
Yeah,
Or
for
yes, actually, see, very, very important. You all are asking very important question. Really, my heart-touching questions. If breeder want to develop a variety for disease resistance, he has to have a good pathologist. In the leadership of the pathologist, we can develop a good disease-resistant variety. If in the leadership of an entomologist, they can develop a variety which is resistant against the insect and pest. But, or if the salinity or aridity, they must have a good soil scientist or a agronomist. Then only they can get. But the breeder people, what they think? No, how their name come in that variety? They will supersede sometime. This is very truly I am telling, and this my perception. Recently I told to that I can develop this variety in those conditions, but I was not allowed.
I also wanted to know, interested that how can the soil scientist can develop a variety?
Yes, I can,
By changing
because most of the variety we are developing in our country, that they are selections only. वर्ण से हम कर रहे हैं, and nature itself a biotechnology. Whatever the natural mutations are there.
Okay, it means for developing a variety, a soil scientist is also not required. Nature can itself develop a new variety, and there it is developing.
So anyone, if I can perceive that, why I am telling soil scientist is not required or required, but those conditions one should perceive, na?
Nature perceives that condition.
Nature perceives. So then let it to the nature. Everything. What is the need for entomologists, soil scientists like that? And that's why nature was moving
No, we need
smoothly.
We need soil scientists, every scientist also, because we want to know what the nature is doing.
Yes,
We have to
nature is doing.
understand.
Yes, that is very
We
much
have to
required.
We have to recognize that variety.
मेरे लिए लोग इतने घबराए रहते हैं कि यार इसे तो कोई किसी से बात करने को डर नहीं लगता है, तो अगर ये डायरेक्टर बनेगा तो खा जाएगा. और ऐसा नहीं है, मैं किसी को खाने और निकलने वाला इंसान नहीं हूँ. कई जगह मैं एग्ज़ाम लेने जाता हूँ यूनिवर्सिटीज़ में, बिकॉज आई एम ऑल्सो PG in ecology and also in that environmental science, and 2 master after my getting the job. मेरा इंटरेस्ट ये नहीं है कि मुझे नौकरी मिल गई. I also got civil services. State I got, and central I have gone under in interview that वो गोल छपा है ना UPSC की, कुरैशी जी के बोर्ड में. But I dropped. My interest was this one, and that time one of my teacher, Dr. N.K. Banerjee, I recall his, and I recall whole of my life. When I was working in the PhD for that my this analysis and all those work, उन्होंने कहा डॉक्टर साहब आप क्यों नहीं इनका हेल्प क्यों नहीं लेते फ्रेंड्स की? सर मैं खुद जानना चाहता हूँ क्या होता है, बिकॉज रिसर्च इज सो इंटरेस्टिंग. जब आप कुछ नई चीज़ लेके आओगे ना, आप बोल्डली किसी भी प्लेटफॉर्म पे आप बोलोगे, तो उन्होंने कहा देन यू आर हैविंग दैट काइंड ऑफ इंटरेस्ट दैट गो आउटसाइड द कंट्री. डू यू नो इफ यू विल रिमेन हियर यू विल बी क्रस्ड, एंड दैट्स व्हाई आई हैव गिवन ए स्लोगन, इट इज़ बेटर टू ड्रेन द ब्रेन रादर स्लेन द ब्रेन. ख़त्म कर दिया जाएगा. हम रतन लाल साहब के पास में थे, ही इज़ वर्ल्ड फुल लॉरिएट एंड नोबेल लॉरिएट. तो हमारा एक जो साइंटिस्ट था मेरे साथ में, वो कहता है अरे ये सर अपने कंट्री में होते तो क्या होता. रतन लाल IRI की MSc किया हुआ है और वो है ऐसे PhD. मैंने कहा इनको क्रश कर दिया जाता, DG नहीं होते. अभी प्रेजेंट DG है, इसको मैंने हैमरिंग की, डॉक्टर हिमांशु पाठक को. ही इज़ हैविंग वेरी एकेडमिक एंड रिसर्च करियर. तो जब वो प्रोफेसर थे IRI में, मैं हैमरिंग करता था, डॉक्टर साहब आप अप्लाई क्यों नहीं करते? मैं तो जेन्युइनिटी और ऑनेस्टी के चक्कर में छूट गया. मैंने कभी इम्पैक्ट फैक्टर इन चीज़ों पे ध्यान नहीं दिया, मैं जेन्युइनिटी पे ध्यान दिया. आपने तो सब कुछ किया, आप क्यों नहीं चाहते? देन ही अप्लाइड एंड CSSRI का डायरेक्टर आज DG है. उसको DDG में सेलेक्ट ही नहीं किया. अरे किसको हटाओ, ये आदमी नहीं यार. और वो कहता था डॉक्टर साहब बड़े झंझट हैं, बड़े झमेले हैं, आप क्या सोचते हो कि मैं कुर्सी बैठ जाऊँगा तो कुछ कर पाऊँगा? मैंने कहा डॉक्टर साहब मैं मानता हूँ कि आप कुछ नहीं कर पाओगे, लेकिन जब तक आप कुर्सी पे हो तब तक गंदगी तो बची रहेगी. तो उसको DDG में नहीं हुआ सेलेक्ट. देन ऊपर भी तो कोई बैठा है, हमारा फेथ होता है. नाउ ही इज DG, ही इज लीडिंग द ऑर्गेनाइजेशन, ही इज अ सेक्रेटरी टू द DARE, डिपार्टमेंट ऑफ एग्रीकल्चर रिसर्च एंड एजुकेशन, and he's a DG of Indian Council of Agricultural Research. He's the apex position. और आज भी वो आदमी वैसा ही है जो हमारे जस्ट हम जब स्टूडेंट लाइफ में थे उस समय जस्ट उन्होंने ज्वाइन किया तो ही इज़ फ्यू ईयर सीनियर टू मी, और आज भी व्यवहार मेरे लिए और सबके लिए ज़्यादातर उनका वही है. अभी दिल्ली गया, मैंने कहा डॉक्टर साहब मैं देखना चाहता हूँ मेरे सपनों का डीजी कैसा है, लेकिन आप ऑफिस में मिले ही नहीं. अरे डॉक्टर साहब मुझे पता चला था आप जैसे आए और वैसे ही मेरे वहाँ से निकल गए मीटिंग के लिए, तो आप कहाँ रुके हो? मैंने कहा नास कॉम्प्लेक्स में रुका हूँ. अरे कि यहीं तो है ना, वो आया उन्होंने घर पे बुलाया. वो वही प्यार वही चीज़ें. तो जो ऐसे लोग होते हैं बाई वर्चू पोजीशन पे होते हैं ना, उनमें मिलावट नहीं होती है. थैंक यू. सॉरी बहुत लंबा डिस्कशन हो रहा है और आपका प्रोग्राम तो थोड़ा अभी और बाकी है. तो हमें मॉरल बहुत ज़रूरी है, और जिसका मॉरल नहीं है साइंस में वो आगे प्रोग्रेस नहीं कर सकता, इवन किसी फील्ड में. ठीक है, सर्टेन लेवल तक हम जाते हैं पर कहीं कहीं हम वी आर द लूजर. थैंक यू वेरी मच.
So,
And if you are having any query, I can discuss, and I will give my phone number. You can talk and discuss anytime. But
we are
time is
highly
limited.
thankful, sir.
Thank you.
Please, Shweta.
Thank you so much, sir. We are incredibly grateful for the opportunity to learn from someone so knowledgeable and so passionate. Your presence is truly cherished. Ladies and gentlemen, my name is Shweta Sharma and I'm your second host for the event, National Seminar on Water, Soil and Biodiversity Conservation, Day 2. Now, we have a very reputed personality amidst us today, Dr. K.C. Soni, Professor, MGSU Bikaner. So I would like to invite Dean, School of Liberal Arts and Sciences, Professor Dr. Jitendra Binwal, to felicitate Dr. K.C. Soni. Thank you so much, everyone. Now, before we proceed further, there's an announcement to make. After our next guest lecture, the session will be open for oral presentation part 1, and at the same time, there will be poster presentation and evaluation in ABB building, 2nd floor, near Dean's Less office. So all the participants of poster presentation and evaluation are requested to contact Dr. Narendra Kumar and Dr. R.C. Nayak with their presentations ready in PPT form in pen drive. Thank you. Now everyone, I'm eager to introduce the next speaker of the day, Professor Dr. Yogesh Joshi, whose excellence, dedication, and perseverance have truly set him apart. Sir's list of achievement is nothing short of impressive. Dr. Joshi belongs to the University of Rajasthan and an alumni from Kumon University. He has a prolific publication record with more than 150 research papers published in various national and international journals. In recognition of his remarkable achievements in the field of lichens and their associated fungi, Sir has received accolades such as the Young Uttarakhand Scientist Award and has been honored as a Fellow of the Linnean Society London as well as a Fellow of the Indian Botanical Society. Sir's ability to consistently raise the bar, and push the boundaries of what is possible is truly admirable. Ladies and gentlemen, I'm honored to welcome Professor Dr. Yogesh Joshi.
A nice morning. Now it's morning to all of you, and it's my privilege that I am here, and I would like to thank the organizing secretary for giving me a chance to deliver a talk. As in the previous session, you have heard about spices, so the session is before lunch, and maybe some of you people have eaten dum biryani. आप में से खाई होगी कई लोग ने दम बिरयानी, तो उसमें एक गरम मसाला पड़ता है, अगर देखा हो, उस गरम मसाले में लाइकेन सी होते हैं, ठीक है? तो is it convenient that I could speak in English and Hindi? Okay. So I am here to tell you about this wonderful word I am taking, because in previous session there are two words sir has told, that symbiosis is there. So I will let you know what is symbiosis. And in your Kulgit, we have talked, I forget the sentence, but it was in Kulgit, निर्माण के पावन युग में वसुधा के कल्याण न भूले. So regarding this Vasudha, we have to conserve our biodiversity. So you are seeing a lot of pictures here, and basically lichen is a fungi, and the definition of fungi you can see is it's an eukaryotic heterotrophic organism, maybe unicellular or filamentous, and have a cell wall of chitin. But now here, anyone who can tell the difference between this? Anyone? No one? This fungi and this fungi. It's a difference or not? It's a huge difference. If you are talking of this fungi with a capital F, then only 4 divisions are there: Asco, Basidio, Chytidio, and Glomeromycota. It means earlier it was Zygomycota. So if your word starts with capital F, then it means you are talking about true fungi, and those true fungi are Asco, Basidio, Chytidio, and Glomeromycota. And if you are talking about this small f, then it will complete all those things. So now the fungi is divided into 3 groups. One is true fungi, and the next false fungi. One is Staminipilla, or you can call it Chromista and molds, and the third one is Protista. You call them as slime molds. So earlier, now you can see here, Oomycota and Myxomycota. This Oomycota and Myxomycota, earlier you used to study in fungi. Now they are Staminipilla and Protista. So this is the actual diagram where you can see these are true fungi. And now in this, along with Oomycota, this diatoms, this brown algae and red algae, they are coming in Staminipilla. So now the algae which you are studying, this brown and red, now they are not algae, they are Staminipilla, and this is Protista group. So why this journey, why this fungi is important? This is the second largest after insects, and if you see, a paper came in the year 1991 estimating there are 1.5 million species of fungi, of which only 80,000 were described. Later, 2 to 3.8 million species were estimated. This much was described. and recently Willis et al. Have reported that there are 4 to 5 million, and recently, before coming last week, one paper came and they said that there are 13 million species of fungi, and you will be surprised that only 2% are described. So the question is, where are these fungi? 98% fungi we have to explore, and where are those? So Liu King, in the year 2017, he mentioned that we have to look for these sites where we can find these missing fungi. The best area is tropical forest, because these are least explored. Second one is the soil fungi of Australia, which we call hypogeous fungi, fungi which resides in guts of beetles and insects. Lichenicolous fungi and endolichenic fungi, I will deal with this here because I am not an entomologist, so I didn't know about all those things. And there are some cryptic taxa, lost or hidden species, and molecular novelties. So what are lichens? It's a very basic question. Anyone who can tell what are lichens? Symbiotic association between algae and fungi. How many are you sure about that? So whether we are using this term correctly or not, and whether it contains some other elements or not, this I will let you know, and I will try to make your concept much more clear. So if we call it symbiosis, then there are 3 parameters in symbiosis. One is mutualist, one is antagonistic or necrotrophic, and third one is commensalistic. In mutualistic, both partners are benefited. In antagonist, one is benefited at the expense of other, while in commensalistic, one partner is benefited but without providing any harm to other person. And these 3 categories are interchangeable. So a species, whatever today is commensalistic, in near future it can become parasitic or maybe saprophytic. As you mentioned that it's a fungal algal symbiosis or fungal algal relationship, then what is the difference between these two? It is also, lichen is a fungal algal association, it is also a fungal algal association, then why we are calling it mycophycobiosis and this as lichen? Here in this case, in mycophycobiosis, mycobiont is living inside and algae is surrounding, while in case of lichens, algae is living inside and fungi is surrounding them. That's a big difference. Similarly, there is 3 biont symbiosis where there are 2 photobionts and 1 mycobiont. We call it bryophilous lichens or cephalodia. When there are 2 fungi and 1 algae, we call them lichenicolous fungi. There is 4 biont symbiosis where 3 biont, 3 algae, 1 fungi, cephalodia, 2 algae, 2 fungi, lichenicolous lichens, 1 algae, 3 fungi, fungi on lichenicolous fungi. Similarly, 5 or more biont symbiosis, then the best example is endophytes or endolichenic fungi. So what happens during these years from 1867 to 1924, we used to see this. Alga and fungi, they meet with each other and propose each other, and they get married and a new thallus is formed. This is lichen thallus. Now you can see neither it looks like fungus nor like algae. And if you cut the section of this, this is the layering where you can see this is called upper cortex, this is algal layer, this is medulla, this is lower cortex, and based on the presence of lower cortex and rhizines, they are the attachment organs here. There is no root system. So before 1867, lichen was considered as an individual. 1867 was the remarkable year when Schwendener mentioned that lichen consists of 2 organisms, one is algae and one is fungi. In 1925, a third partner, that is bacteria, came into existence. In 2011, along with bacteria, this is the partner you can see, secondary algae is there. Now they have 4 partners. In 2015, Protista came. In 2016, another algae came. And in 2019, now you can see virus is also there. So in a lichen, you have algae, fungi, secondary fungi, Protista, virus, single cell yeast. These are the things. And if you see this, in this portion you can see this is the cortex, algal layer, medulla, lower cortex and rhizines. Now as far as composition is, you can see this upper cortex is made of fungus, this medulla is made of fungus, this lower cortex and rhizines is made of fungus, only this much portion is of algae. So now what we should call, as you have said that it is a symbiotic associationship, then it means both partners should be in equal amount, 50-50. But here, in case of lichens, 90% is fungi and 10% is algae. If algae is going to jump from 10 to 10.1% or 10.2%, this fungi will kill that algae. It will not allow to raise him. So it can't be a symbiosis. So what is it? Second thing, we can call it as a controlled or balanced parasitism. It is a type of symbiotic association, but we have to tell it that it is a master or slave relationship where master is fungi and slave is algae. And if you know this ICN AFP, it means International Code of Nomenclature for Algae, Fungi and Plants. Here algae is written with small, F is written with small and P is written with small, because it has to incorporate all the algae, all the fungi and all the plants, which I have tell you in the previous slide. According to ICN or ICBN, whatever you want to say, this lichen did not follow Article 13.2. Article 13.2 of ICN used to say that each and every individual has a single name. So if a lichen has two organisms, one algae and fungi, we have to give name to both of them. But we are giving name to a single partner, and that single partner is mycobiont because it is contributing 90%. So it's better that we should call them lichenized fungi. And this paper is being published in Science, where yeast has come as a third important partner. So now the actual definition of lichen is that it is a fungus or fungal-like organism where the primary mycobiont surrounds the algae from outside, and they have some other important microbiomes associated with, like fungus and bacterial microbiomes associated with them. And you can say that now people are saying that they are analogous with agriculture, because in this fungus platform is providing algae the space and the algae is producing food, just like agriculture fields. So if you are using this word in case of taxonomy, then you have to use the word lichen forming fungi or lichenized fungi, not lichens. So these are important. You can see places where you can found lichens. It's on the bark, it's on the twig, it's on the soil and leaves, rock, and this taken from Kedarnath. After that tragedy, the animals and human casualties, the bones were left, and then after some time these bones were full of lichens. Similarly, during my stay in Uttarakhand and even in this Amer Fort and Nahargarh Fort, you can go and visualize the monuments that they will be full of lichens. This old car, this is churchyard where you can see the fences. And once again, in Current Science, I have published a paper. This is a nylon net house which is used in farming. It is wholly covered with lichens. This is the plasticolous lichens which we have reported. These are the basic techniques and basic instruments which you require for identification of lichens. And in this is a TLC jar where we used to perform the TLC, and this is the UV spectro cabinet box. Because some lichens, later I will tell you that lichens are a source of secondary metabolites, and they used to produce 1,000 secondary metabolites which are not produced by any other group of plants. So this instrument is required to analyze whether they are UV plus or UV minus. So there are some lichens which used to grow in Badrinath, Kedarnath, which can be used as UV protectant cream. So these are some instruments which we require for identification. This is the general process. And these are basic some chemicals which we need for their identification. Now these are some peculiar features about lichenized fungi, that they are ubiquitous and perennial. They lack cuticle, because of which they have the tendency to absorb the material, as in the previous lecture sir has told that about bioaccumulators. So these are the best bioaccumulators, and recently a paper has been published that they can accumulate arsenic from the rice fields. So they have a controlled parasitic nature, they are poikilohydric, they lack the mechanism of active absorption and excretion, they are photosynthetically actively throughout the year. They are the best bioindicators. Why they are the best bioindicators? You can see here, because sir has also told that don't rely on technology. So you can see here, if you go to the field and use lichens as a bioindicator, then these are the benefits which you can have. There no need of equipment is required, no methodologies, there is no reason for theft and vandalism. This is very much economical, handling process is easier, no electricity is required in comparison to instruments. And this is a lichen community, you can say. What it will tell you, if you go to a forest and if you find this type of lichens, pinhead lichens, then you can definitely say that it is an old growth forest. If you find parmeliod lichens in a forest, you can say that anthropogenic pressure is there and the forest has been thinned out. If you find this type of lichen, then the atmospheric air is very good, you can live there. If you found this type of lichens, then the nitrogen content is enormous in that forest. And if you find this type of lichens, then go and build the house. That area will not have any volcanic activities. It's always grown on hard rocks. And similarly, by performing this experiment, you can define your areas, because if I am living in Jaipur city, in Jaipur I will not find any lichens, then it is, we call it as lichen desert. When I move from like Jaipur to Amer, I found a small piece of lichens growing there, then it is in a struggle or transition zone. Moving ahead towards Kotputli, I found a lot of lichens and bryophytes. It means now the environment is good in comparison to Jaipur. The air is fresh. So effects of climate change, if you see on lichens, what I have observed in Uttarakhand while I was in Uttarakhand, that majority of thermophilous lichen species which used to grow in the tropical regions are now shifting towards Himalayas. So these species which are in tropical regions are now in Himalayas. Another important thing is that this folicolous lichens, there is folicolous lichens which used to grow in tropical rainforest, and they are the bioindicator of tropical rainforest. If you go to a tropical rainforest, you will see the leaves and you will find a lot of lichens growing over leaves. You can definitely say that it is a tropical rainforest. These lichens which used to grow in tropical rainforest are now growing in Himalayas. It means the climate of Himalaya is changing. Similarly, chlorolichens have increased in reference to cyanolichens. Cyanolichens means those lichens which contain cyanobacteria, it means having the nitrogen fixing capacity. Now they are declining in Himalayas. So these are some pictures of these thermophilic lichen species that I have mentioned you, that these folicolous lichens, these are in Nilgiri, in Palni, Andaman, Nicobar and northeast India, and now they are switching towards Uttarakhand. And this is the plasticolous lichens, as I have told you about this. Now the question comes, because this needs to be worked out, since they are growing on plastics, can they be responsible for plastic degradation? It's a big question. Till date, nothing. Because lichens use plastic as a substrate, it did not take anything from the substrate. That's why it cannot degrade plastics. So these are some tolerant or toxic-tolerant lichen species. The arsenic accumulation experiment has been done on these Phaeophysia, Hispidula, Orbiculus, and Pexinococcus. And Pexinococcus is the most pollution tolerant lichen. So this is the monument. You can see it's covered with lichens. And now what happens is that in 2001, Nimitz said that monuments can act as ecosystem because lichens and other plants are growing on it. And as per IUCN Conservation Congress, what they said that the monuments are placed in category 3rd and we should protect them. Now it's up to you that if a lichen or a plant is growing on monument, it is an ecosystem, how you feel, whether you treat it as a curse or a boon. To me, it looks beautiful, but for you, it won't look beautiful. So it's up to you. These are some lichens of Rajasthan. These are some new species. It was described by me, abuensis from Mount Abu, Avasthyae, and these are some others. And these are some new records from Rajasthan. And this is important thing is that Hepia, Feliscum, and Thyria as well as Zal Bracknella, they are endemic only to Rajasthan. They are not reported from anywhere else from India. These are endemic species. Now the second part, that lichens contain some other microbiomes, and I am dealing with fungi, so I will tell you about which sort of fungi they used to contain. But before that, you know these three words: phyllosphere, rhizosphere, and bryosphere. Phyllosphere means, anyone? Phyllosphere? Area surrounding the leaves. So whatever the microbe around the leaves is phyllosphere. Rhizosphere, microbes around the root, and the bryosphere, microbes in and around bryophytes. Now, whether something around lichens or not. So this is a question. For this term was given by Santiago in 2015, lichen sphere. The surface and narrow region of the lichens thallus that may represent a protected natural microhabitat with favorable conditions which can help non-endemic, non-lichenized fungi and other life forms to survive and disperse in extreme conditions. In India, lichens study has started in 1950s, and till 2013, nothing was known about these secondary fungi. So I got the privilege when I was in South Korea to learn about these, and then I started this journey. And till date, I have described a lot of new species on this lichenicolous fungi. I will let you know. So why the problem, why there was a decay, why there was a delay in studying this fungi? Because of these two reasons. Lichenologist, the person who used to study lichens, they don't know anything about mycology. And the mycologist, they never used to collect lichens, and they don't know the terminology of lichens. This is a big problem. Now, you can see all are same. Anyone? All are same? Looking same? No, except this one. All are Xanthoria elegans. This is also Xanthoria elegans. The color is changed a little bit because it can change from yellow to orange, orange to yellow. That's the only thing, growing in Badrinath area. Now, if they all are same, then what's going on? The lichen is same, but you can see here the black dots. This one, this black dot, this, and this. A single lichen is having 1, 2, 3, 4, 5, 6. 6 different species of microbiomes associated with it. A single lichen. So this is Arthonia, this is Mirrella, this is Bellemirella, this is Stigmedium, Cercidospora, Mirrella. A single lichen, and that lichen is Xanthoria elegans. And the peculiarity of this Xanthoria elegans is that this is yellow-orange color. It has anthraquinone, and in near future, if scientists try to, if they can isolate this anthraquinone, then it can act as a UV protectant cream. This is the best thing about this. So this is, you can see now. Everyone knows very common lichen, you call it Parmelia, Parmelia, Parmelia. So it is Parvotrema, a member of Parviliaceae group. This is what I have observed in India is that, sir, we want to do bioprospection activities. Okay, send us the material, you give us the material, and we screen it. But the problem was that we never looked into the lichen material carefully. Now, this entire is a very healthy lichen, nothing is there. Okay. Now see. Did you observe something? No? See these arrows carefully. Small dots will be there. I have zoomed it, and when I cut the section, this is there, the hidden microbiome on the surface. So these secondary fungi can be categorized into these groups: saprophytes, parasites, parasymbionts, and mutualists. And these parasites are further classified into pathogens, necrotic spots, and gall-like malformation. This was a definition given by Hawksworth, and recently we have modified it, and we say that now the secondary fungi, as per Hawksworth, this much is okay. We have added a second category also. This is endolichenic fungi, just like endophytes, which you isolate from other group of plants. These endophytes are also residing within the lichen thallus, and we call them endolichenic fungi. So there are two groups of fungi which used to live in or on the lichen thallus. If you see this, if you consider my hand as a lichen, then the fungi which are growing on the surface and which you can see with your eyes or microscope, it is lichenicolous fungi. And the fungi which are living inside along the medullary portion, they are called endophytes or endolichenic fungi. So I welcome you to this world of, we call them as exploiters of two kingdoms, because this fungi, lichenicolous fungi, which is growing over the surface, it can kill both algal and fungal partner of lichen. It means it can kill lichen. They are invaders, they are niche seekers, and they are takeover specialists. How? As I mentioned, they are saprophytes. On dead lichen thallus, you can see a lot of fungus growing, and this is Aspergillus is there, Cladosporium is there, Penicillium is there. It's on the dead surface of lichen thallus. Acrimonium lichenicola was the first saprophyte reported. Now, as I told you that they are pathogens, they can entirely kill the lichen thallus. And if they kill the thallus, it means they are going to kill themselves also. Because if their lichen thallus is not there, these will not be there. So, Ethelia arachnoidea. Sometimes when they used to infect the lichens, then you can see here these black dots is restricted to one or two positions like this. So small portions get discolored. When this infection is severe, now you can see it's a healthy apothecia and this is a damaged apothecia. Sometimes what happens is that you have seen a lot of leaves infected with galls. You have seen gall-like structures on the leaves. Similarly, in case of lichenicolous fungi, there are some species which used to form galls on the thallus of lichens. And this was the first lichenicolous fungi reported from the world, Biotropsis asnierum, which used to form galls. And these are generally members of Basidiomycetes fungi. Sometimes this was the first lichenicolous fungi which I identified when I was in South Korea. It is a parasymbiont, or we can call it as a common cyst, in which a fungus grows on the thallus of lichen, and the algae which is making food for the fungus also makes food for this another fungus. They did not cause any damage, they live smoothly there. However, in case of mutualist, what happens is that a fungus comes and replaces the original fungus and takes over it. Now, during this my entire journey, what I have done is that this is the first new genus for India as well as for the world, which I have described, published in JAPB. This is Ajaysingha dendistrictae. You can see some gall-like structures. These are not galls, but a stroma, and in stroma you can see the spores and perithecia. Second one is Jejulia bisolomoids. This is from Jeju Island in South Korea, from where we have identified this. You can see hyaline transeptate ascospores in it. Now, this is Pertussaria, this is Pertussaria. The host is same, but here you can see in this host a single perithecia is there. However, here it is a stroma. So this is Polycoccum aquarianum and this is Polycoccum hawksworthianum. Two different species, the host is same. Again, this is Pertussaria and Ochroleutia, the same host, but here you can see on the apothecial disc margin there are some black dots. These are Coelomycetes members. When you cut it, then you can see the conidia. So this is Lycondiplis ochroleuciae. Again, this is a Pertussaria. I am telling about only Pertussaria. Now you can see 4 species have came. Now in this Pertussaria there is slit-like appearance, and when you cut this slit, you can see these are spores, and this is Labrocarpon submuriforme. This is the first time that in a Labrocarpon we have found submuriform spores. This is Cercidospora neveroi. You can see this is the healthy apothecia, and in this healthy apothecia, in and around healthy, these are some small black dots. When we have cut these black dots, we found this is species. These all are new species. This is from cold desert of Ladakh, where you can see this is a lichen called Lasidia, and here these are some black dots. When we have cut these black dots, these spores are there. These all are published in Syrovia, Phytotaxa and other journals. This is in Q bulletin, again from Ladakh, this material, and these all are stress tolerant lichens, and here you can see a black dot. When we have cut this black dot, this spores are there. This is Zooxiomyces protoparmilopsis. This is Lichenocora ajacingii. This material was from Philippines. A person has sent me this material, and when I visualized it, I found it that it is another Lichenicolous fungi. This belongs to genus Ophiographa and published in Q bulletin. Now here there are 2 different species. I will show you this one and the previous one. This is Sclerococcum. The next one will be Sclerococcum. The difference is that here in this Sclerococcum you will be seeing the sexual stage. Here the apothecia are there, while in the next one you are seeing the anamorphic stage, the wicks-like conidia. This is again Polycoccum lecanorum, new species from Ladakh. Toninia also from Ladakh. Pyrenedium is from Madhya Pradesh. So these are some additional records that's being, I'm not going in detail about all those things. These are some, you can see it's a lot. Mainly these Lichenicolous fungi are confined to alpine regions of Himalayas. That's the beauty of this. So what's the problem is it? It's finding a needle in the haystack. If you ask me to search a needle in this entire room, it will be very much difficult for me. That happens with lichens also, because lichens has this much size, and we have to search for a drop or a pin in that. And a person cannot be expert in all the group of fungi which Lichenicolous fungi having, because Lichenicolous fungi belongs to Asco, Basidio, Silo, Hypho, and a person cannot be an expert in all those groups. That's a problem. And why I say that looking for a needle in a haystack, material was sent to me that please perform the antimicrobial, antioxidant activity of this, and it's a very healthy sample. I said okay. Then I started looking on it, and maybe you are saying that it's a healthy thallus. On a single portion here and here, what I observed is this. Reticulate venations, and when I cut, I found spores. It is again a new species. Now the second part is these endophytes which are residing inside the lichen thallus. So you have to culture them, and this term was given by Mirdliskova, and the best beauty of this is that you have to go to a forest, collect lichen and come to the lab, and just by sitting you can do the experiment, nothing else, not tedious one. And this is the same thing. So this is the process how we used to do it, and this is the representation of that in the form pictorial representation. You have to surface sterilize the lichen thallus and then place it on the media, and then the colonies start coming after 3 or 4 days and start with the help of manuals you have to identify them. And the beauty of this endolycanic fungi is that they produce enormous secondary metabolites, and of these secondary metabolites you can see until date 351 secondary metabolites are known, of which 196 are novel compounds, and all these have antifungal, antibacterial, antiviral, Alzheimer's and anti-HIV activities. These are the compounds which have been isolated. These are all novel compounds from endolycanic fungi, and this we have published when I was there. With my student, we have published a book called Endolycanic Fungi Present and Future Trends, and we have isolated a lot of endophytes from it. So these are some images of endolycanic fungi. You can see here Alternaria, Aspergillus, Phytomela, Cladosporium is there, Pistolopsis is there, Sphagezinia is there, Absidia is there. So these used to come frequently. So at the crux you can see that when these 3 categories you consider, it was the Linnaeus who for the first time reported lichens from India, and then Lichenicolous fungi was again reported by European scientist Morio in the year 1951. However, as far as India is considered, Indian workers are concerned, it was Qureshi who identified lichens for the first time, and then Avasti and Singh started Lichenicolous fungi. The milestone, a turning point come in the here. You can see lichen started 1948, it's me, and here it's me. So if you see here that the number of new species, percentage of new species described from India as far as lichens are considered, it was 10, and here it was 12. The ratio is now increasing here. So it's the conclusion part that lichens, it's better you should call them lichenized fungi. And if you want to discover for a drug, go for a drug discovery, then I would like to suggest that go for endolycanic fungi, not for lichens, because the biggest drawback with lichens is that a 1 mm thallus of lichens need approximately 5 to 10 years for its development. So it's better take a small fragment, isolate enormous number of secondary endophytes from it, and mass culture those endophytes, they will give you better result. Second thing is that if you want to study the co-evolution of these fungi, they can act as a model organism. Third important part is that these black fungi or Lichenicolous fungi, as they used to grow in the most stress resistant environment, that's why they are called as most stress resistant organisms on earth, and they are a part of astrobiology. And recently a paper has been published that lichens are surviving in Mars. So now people are taking lichens along with them to space for studying the behavior, what's going on there. And these specimens, these species, Aspergillia, Buellia, Circineria, Rhizocarpon, Russalskia and Xanthoria, they all are space fungi. And of these space fungi, Rhizocarpon and Xanthoria paratina and Russalskia elegans, they also occur in India. So if a person, if Indian persons, ISRO scientists want to do research, they can take these with them. So I would like to acknowledge my teachers who taught me this, and to students because of which I am here, and these funding agencies who supported me. So I request lichenologists and mycologists should join hand, then we can resolve the problems. Thank you very much. Any questions? Yes, please. Yes, sir.
Namaskar, sir.
Namaskar.
Sir, lichen grow in many of the areas, many of the soil conditions. Just I want to know, can we develop any lichen that can be used as a food and nutrition?
Sir, you cannot culture lichen. That's a big problem. Second thing, lichen, there are several lichens which are used as food and spices. I have not taken that part here, but there are enormous. If you go to Japan, they will give you Cetralia boiled in water so that all the acids are removed. You can have it as a salad. Like reindeer moss, you have known, Cladonia. It's being eaten by insects, animals. So there are several lichens which are being eaten, and even in India, the garam masala, it's being eaten. But the problem is that we cannot cultivate or grow lichens. It's a damn sure fact, because they have two partners. If you are going to culture them in laboratory, what you will do, you first of all isolate fungi and then algae. The nutrient media of algae is different and that of fungi is different. One is for liquid media and one is solid media. You cannot culture. That's a problem. Anything? Yes. But nature? Sir, in nature, as I have told you, that they did not take anything from the substrate. They just adhere to the substrate. They will not take. They take mechanical. The algae is providing food in the form of mannitol, and that's being utilized by the fungi. That's a beauty, sir. Yes.
Sir, good work.
Thank you, sir.
Thanks to see the transfer of legacy from BL Jain to Sudhakar Mishra to Yogesh Joshi now.
It's my pleasure.
So now you have become pioneer in this field. And
Sir, I cannot say pioneer. I am still learning
in India.
Not in India because
most of the scientists are not working on lichen here in India.
In
only
India,
few scientists are working.
Only few scientists are working, and that's been confined to laboratories, CSIR, NBRI, they are working. But in university system,
nobody's
no,
working.
nobody's.
Yeah, that's why I called you pioneer legend.
Thank you, sir.
Sir, I have a question. Sir, you called it multi-partner lichen association. Can we call it colonial nature, because they are all these partners are working differently, and they have their own all of them have their own role in that.
Sir, you are absolutely right. And in that case, what happened is that now lichens are called as ecosystem. Like as you said, communal partner.
Colonial.
You are saying colonial partner. Now recently, Hawksworth, they have revised the definition and they said that lichens are an ecosystem. Because they are interacting with non-living substrate, but have living partners, lot of living partners within them.
But they are part and parcel of the ecosystem. We all know.
Yeah, that's the thing.
Thank you.
Sir, thank you very much. It is your very specialized and very informative presentation. One of the question already asked that I was willing to ask that what is the economic importance. The second question to remain, and that is, okay, we cannot use them, we cannot culture them, but at least you told that climate change impact or this and that, these are the indicators.
Yes.
So how many these lichens are there? By that way we can, these are called as the bioindicator for certain adversities or abnormalities.
Sir,
and how do we harvest their this bioindicator?
I will show you, sir. Sir, this is the list. Okay. You can take any, there are two different approaches what you can do. You go to the forest, take a piece of lichen, place it in a cardboard and hang it at the crossroad, wherever you want to know the pollution level. Hang within 1 month, 2 months, 3 months, take that lichen, scrap it, go for the metal estimation. It will let you know how much metals have been accumulated and what sort of metals have been accumulated, it will tell you. That's the best part. Second part is that go to the forest, or maybe you can do one thing is that in the laboratory you have bring, place it in a cardboard, make solutions of metals, spray them, after 1 or 2 months, analyze. The best part is that lichens lack cuticle, so whatever you are going to provide it, it will absorb. Drawback with machine is that if you take the machine right now, it will provide the reading of today, but lichens are accumulating it from past. That's the best thing.
Very good. Nice. Thank you. Thank you so much.
Any further questions? Okay.
Thank you, sir, for sharing your remarkable knowledge with us. Before we move ahead, there is an announcement. All the participants of the oral presentation session are requested to provide their PPT presentations in pen drive form to Dr. Narendra Kumar and Dr. R.C. Nayak. Thank you. Now, it is a matter of tremendous delight for me to invite our next speaker of the day, Professor K.C. Soni. Born on 26th of January, Professor K.C. Soni has about 34 years of experience in teaching and has published 40 research papers in various journals of national and international repute. Professor Soni contributes regularly to professional literature, develops innovative institutional methodologies, and is committed to the conservation of science, which is crucial to meet out the needs of the society today. Moreover, Professor Soni is an avid photographer, and many of his photographs of wildlife are regularly published in magazines and newspapers. Professor Soni has also developed over 300 video lectures on zoology, which can be watched on his YouTube channel. Now, I would like to invite the Honorable Professor K.C. Soni on the stage for his lecture.
Hello to everyone. This work is a part of minor research project funded by University Grant Commission. My one student, Mr. Chandrup Soni, who is pursuing his PhD work on this animal. My today's talk topic is biodiversity analysis of Egyptian vulture, Neophron prochnopterus, in and around Churu city, Rajasthan, India. in India, we can find two species of this vulture. One is Neophron Prachnopterus, and second is Neophron Prachnopterus ginginianus. Among this, one is Indian species and one is migratory, that is from Europe. Indian species you can see throughout the year, but European species you can see from month of November to month of February. इसमें प्रोसीड कैसे हो रहा है देखना है. ओके ओके, हाँ, नो प्रॉब्लम. This one is Indian species, which have yellow bill, weaker feet and claws. This one is European species, in which the bill tip is black. So if you go in nature or in wild area, forest area, if the bill means beak, bill is pinkish, then it is Indian species. A bill tip is black, then it is European species. Lots of work has been done throughout the world, mainly in Spain by Bergier, Ceballos, Donajar, Israel, former USSR, South Africa, Italy and Vienna. Very little work is done in India, but especially in Saurashtra region, University of Rajkot, Professor V.C. Soni and his students did a lots of work. In Rajasthan, Vibhu Prakash from Jodhpur University and Dr. Anil Changani from NGS University, Bikaner, did a very little work. But still, the entire history of this world is to be explored. My study area is in Churu city. In this figure you are viewing, Churu is located 28 degree 15 degree north latitude and 74 degree 50 minute east latitude. Churu is a part of arid zone. In summer temperature goes beyond 52 degree and winter temperatures go beyond -5. So we have both extremes. In winter season we face -5 degree and in summer season we face up to 54-55 degrees. And summer wind blow very hot, that is called loo. And winter the wind blow very freezing, that called damphy in local language. In summer season you can face loo and winter season you can face damphy. These are some local plants, because every bird when breeds they use local flora for the construction of their nest. So here herbs are present, shrubs are present and large trees are present. The main herbs of this area are bui, cholai, pili katili, bharut घास, bathua, sania, दूब, मोठ, दुचावड़ी, देदी दुधेली, चिड़ी बाजरा, गरमानो, पानी और कुंचा, बांसा and bhakri. Among main herbs are fog, akda, केर, धतूरा, खीप, लाल कनीर, पीला कनीर, and two types of boar, Gigipus jujuba and Gigipus numularia. Among main trees are kikar, sares, neem, sisam, gulmohar, safeda, bargad, पीपल, खजूर, जांटी और खेजरी, विलायती बबूल, जाल, फ्रांस and रोहिडा. In Churu city, Jhunnu city, Sikar, hilly area is very less. Although in Jhunnu, Ravli hills we can see. In Sikar, if you go to Hers, just 30 kilometers from here, you can see mountains. But in Churu city, there are no hilly area. But there are so abundant Havelis built by multi-millionaire state peoples, and each Haveli on outer side they are supporting balconies with todas. In this Haveli you are seeing todas. So these birds use this area as breeding sites. In every Haveli, Egyptian vulture breeds very successfully. Although by IUCN it is listed as endangered bird. But in Churu city, and even though in my town, I belong to a very small town, Ratanagar, just 40 kilometers from Lakshmangad, and there are about 50 Havelis, big Havelis, and each Haveli one Egyptian vulture breeds every year. So success rate of breeding is very, very high. But unfortunately it is listed in endangered species by IUCN. I discussed on 3 or 4 aspects about its population and roosting, then its foraging, then its breeding. Breeding part is main concern of this. In this picture, you are looking 2 adult and 2 sub-adults of Egyptian vulture roosting on a khezri tree. 1 adult and 2 sub-adult depicting day roosting on vertical stone bars in forest area. Single adult Egyptian vulture exhibiting night roosting on electric pole. Single Egyptian vulture showing roosting on an old Haveli. 5 Egyptian vultures showing roosting on high tension electric line. 1 Egyptian vulture roosting association with black ibis on bare tree. 1 Egyptian vulture and little egret showing roosting association on the boundary wall of cremation center, and roosting association with other species of vultures on Jal. These are some populations data of Churu city, about 40 to 50 birds. You can watch anytime. Among foraging, it feeds on a variety of feeding materials, like 2 Egyptian vultures at the site of dog carcasses, immature Egyptian vultures with black ibis and house crow. They are feeding on sarcophaga fly. An adult and juvenile feeding together at dead body dumping station. A juvenile feeding on cow carcasses. In Shraddh Paksh or Navratra Paksh, we generally offer puri and gulab jamuns, jalebis to this bird, and I communicate this finding to publish in a very famous journal from Africa, South Africa, Vulture News, but they rejected my paper, and they warned me that this is a scavenger bird, this is carnivorous bird, and why you are offering them sweets. And this news is published in local newspaper Rajasthan Patrika कि vulture को भी भाने लगे हैं लजीज व्यंजन. But my paper, that paper was rejected, and they strictly warned, you should go to jail. When they are scavenger birds, they are carnivorous birds, why you are offering them herbivorous food material or sweets? But it is real observations. During Shraddh Paksh or Diwali or Holi, we provide them puri or any sweets like gulab jamun, rasgulla, jalebi, and these birds feed very naturally. Egyptian vulture feeding on human excreta. because still defecation facility is very poor in rural area, and persons go for defecation in their forest area, their grasslands, and this would feed on human excreta. In graveyards, they feed on small bones. After burn of human small bones, they also show bone eating behavior in graveyard area. It is a real observations of Sardar sir. I appointed as a in election during this November, 23 November, and I observed that one pair of visitor vulture killed the hedgehog in natural habitat. So I photographed them. This is one pair. They kill the hedgehog. And at last they take in beak and flew away towards their nest. Now this is the breeding part. It is the main presentation of this PPT. Breeding part cover courtship behavior, copulation behavior, nest building behavior, incubation of the egg, hatching, then care of their nestlings, care of their fledgling, and some observations on post fledgling behavior. Courtship behavior is started in the month of October and November. Pairing is started. Male and female just close together, sit together on nearby trees, nearby plants, nearby havelis. This is, in this picture you are looking pair bonding prior to courtship. This is a courtship display, a silent posture of courtship display. As the courtship duration proceeds, they comes close together, and they are taking sun bath in the month of November. Sun bath during courtship period. As close bonded posture during courtship display. When pair formation is permanent, they start beeling and kissing events. Beeling events prior to copulation, beeling and kissing event before the copulation. Sometime extra pair copulation is also observed, EPC. The male bird is ready for mounting on female. The first step of mounting, and bird start the cloacal kiss, because in birds there is no penis or vagina like genitalia. So they start cloacal kiss to sperm transfer. The female remains silent and male start wing flapping and produce grunting sound. Wing flapping speed increase and pitch of voice rises. The middle stage of copulation and frequency becomes slower down. The post-middle stage of copulation and cloacal kiss now completed, sperm is transferred, and this is last stage of copulation, and male again produce loud voice. In birds I observed, in eagle, male partner remains silent but female produce voice. But in Egyptian vulture, female remains silent and male produce voice during copulation. Male bird now in mode of flew away from the mounting site, and copulation event is now over. Both male and female flew away from the site, and they start second copulation event in nearby area. After copulation event, they start nest takeover and egg laying, and you are seeing in this picture two sporting balconies of two havelis of todas. So they now start built up nest. Sometimes in one haveli, different todas. In one haveli of Kedia family, 5 pair of Egyptian vulture breed every year in separate areas. So it may be colonial rooster or it may be social. you can consider this event as a colonial or social. Sometimes they also construct nest on khejri tree. So this is a nest on khejri tree in Dhani Lalpura, just near to Churu city. Nest on khejri tree made from sticks of local flora and lined with fur, cotton, rag, leathers. Male Egyptian vultures with nesting materials. They carry cottons of hospitals, polythene, rugs, hair, cloths. Now they started nest construction. A male bird carrying cotton for build-up of soft pad in nest. A nest of Egyptian vulture with egg having cloth. In almost all nests, I found a red cloth. Red cloth may be an indication of occupied that area for nesting, or it may be for warning signal also. So in each nest, you can find a red color cloth of red color or red ribbon, which indicates that this area is occupied by our partner, and it may be warning coloration. This one is egg. Generally one egg is laid in each nest, but sometime two. So clutch size is 1 or 2. Size of egg varies from 7 to 8 centimeter length and diameter 4 to 5 centimeter, and its weight is about 50 to 60 gram. First egg is bigger in size with more weight. Second egg is smaller in size with less weight, 5 to 6 gram less. Incubation period 45 days. During night only female incubate, but in daytime both male and female incubate after regular interval. From rising of early in morning 7 a.m. female flew away for feeding in forest area, then male incubate, then female comes and male go for feeding. So in day they regular change their duties from every 2 hours, but in night only female incubate the eggs. And after 45 days hatching occur. Sometime female is very this is photo of Lalpura Dhani. I start to photograph this bird, but female raise their feathers just for warning. But males are always promiscuous. They never protect their nest. They flew away. So we plan a guideline when male birds are sitting on the nest. A female is in field area. So I take my photographs only when male is sitting on the nest, because female is threatening. Sometimes they attack. Female Egyptian vulture incubating the egg in nest. Egg of Egyptian vulture in the nest showing hatching, and in this picture you are looking a very small immature nestling. Female Egyptian vulture brooding the blight naked just born nestling, and after this every 4 days I take photographs. We hang a big mirror on the nest from roof, and we take photograph from the image of nest, because otherwise it is very difficult to photograph, because Havelis are 30 to 35 feet height, and build up of machan is not possible. So we go on the roof of that Haveli, we hang a big mirror in front of the nest, and then from camera we take pictures from the image which we seen in mirror. This is 8-day-old nestling. Their eyes is clawed, down feather creamy in color, nostrils on beak. 12 days old chick of Egyptian vulture with wing buds, tarsus developed and bill further developed. This is close-up of immature nestling. 20 days old nestling of Egyptian vulture showing remarkable growth in head, bill, feathers and tail region. 28 days nestling depicting further growth in all parts of the body. 30 days old fledgling showing flapping of wings. When these immatures start their flapping of wings, then it is called fledgling, not nestling. So starting period is called nestling growth, and when they start flapping of the wings, that is called fledgling growth. So entire duration is divided into two parts, nestling period and fledgling period. And you are still seeing the changes in color in feathers. Starting stages are creamy in color, and now blackish pigment, brownish pigment start in their feathers. Some interesting observations I also observe. Suppose one egg is laid by a pair that hatch, and nestling attain a growth of 30 days, then female lays second another egg in same nest, same female same nest. तो एक अंडे से बच्चा निकल गया, बच्चा 30-35 दिन का हो गया, उसका दे वही फीमेल दोबारा फिर एग लेइंग करती है. This is asynchronous breeding. It may be due to changes in sudden climatic conditions, because recently during Holi month temperature goes 45 degrees, then again ice crackers, rainfall, temperature again down. So very fluctuations during 15 February to 15 March we have to face, and in these conditions they show this asynchronous breeding behavior. In this picture you are seeing पहले अंडे का जो बच्चा है वो पीछे खड़ा है, और सेकंड जो एग दिया उसको फीमेल इनक्यूबेट कर रही है. और इसमें ड्यूटी जो है बहुत हार्ड हो जाती है. इसमें फीमेल 24 घंटे अंडे को इनक्यूबेट करती है, और इसका जो मेल पार्टनर है वो इसको फीडिंग भी करवाता है, बच्चे की फीडिंग भी करवाता है और अपने फीमेल पार्टनर को फीडिंग करवाता है. This is 40 days old fledgling depicting clear line pattern of feather. 45 days old fledgling showing black feathers in the neck region and further developed rectrices. This is 60 days old fledgling sitting on the nest, and 65 days old fledgling sitting in the nest. ये हवेली है केडिया परिवार की, हमारे गांव है रतननगर जिसको केडिया परिवार ने बसाया था. तो उनकी ये बड़ी हवेलियां हैं, और एक हवेली में 200 टोटे हैं. सेठों की भी क्राइटेरिया है, सेठ किसको बोलते हैं. आप पुराना लिटरेचर यदि पढ़ो तो सेठ वो है जो हवेली बनाए, टोटों की हवेली बनाए. सेठ वो है जो वाटर बॉडीज़ बनाए, जोहड़े बनाए, बावड़ी बनाए. सेठ वो है जो मंदिर बनाए. सेठ वो है जो धर्मशाला बनाए, सराय बनाए, टेम्पल बनाए, और गरीब बच्चों को रोज़ाना भोजन कराए, और मरने के बाद में अपने ऊपर छतरी बनाए, सैनोटैप. तो ये 10-11 गुण है जिसमें होते हैं, उसको सेठ बोलते हैं. पैसे वाला कोई भी हो सकता है, लेकिन सेठ के लिए यहाँ का जो लिटरेचर है उसके अंदर ये क्लियर कन्वेंशन है कि सेठ किसको बोलते हैं. और वी आर वेरी फील वेरी प्राउड दैट Shekhawati is very rich in about 300 families, and 70% economy of India is governed by this Shekhawati region. Ruyas are here, Modis are here, Todi here, Kedia, Podar, Jalan, Singhania, Ruya, Tosniwal, Rungta. 70 days old fledgling in standing position having blackish brown coat color. 80 days old fully developed fledgling of Egyptian vulture having black feathers in their head region, and now they have well developed tarsus, toes, and rectrices. Fledgling of Egyptian vulture sitting on the wall of Toda before first flight, and after hatching this complete cycle completed into 80 days, 40 to 45 days incubation and 80 days further growth. So total development duration covers 120 days. This life journey is about 4 months. One breeding success take 4 months, and 1 or 2 months in courtship event, copulation event. So this entire journey completed in 6 months. You are seeing this picture, 80 days old fledgling after leaving the nest, sitting on the roof of an old haveli. This is first flight. And after flight, fledgling remains with their parents, generally with their mother, and they learn how to feed, how to tear the carcasses, how to protect, how to fly. So they learn 3 to 4 months, and they remains with their parents. This is demanding feeding behavior. Fledgling is demanding for food from his mother. Sometimes due to the use of insecticides, pesticides, diclofenac sodium, meloxicams. So in winter season, daily 1 or 2 cattle's died, cattle's, buffaloes, and sometimes both pair goes for mortality due to this insecticide use, pesticide use, diclofenac use. So this is observation of Chandgotia Haveli, and both partner, both male and female partner, died in their nest. And इसमें एक अंडा भी था, फीमेल इंक्यूबेट कर रही थी, लेकिन फीमेल की डेथ हो गई, 1-2 दिन बाद में मेल की भी डेथ हो गई. और इसको फिर मैंने डेड बॉडीज़ को उतारा, इसका फिर पोस्टमार्टम किया, इसका पूरा स्केलेटन है वो भी हमने तैयार किया, वो हमारे जूली म्यूजियम के अंदर ये रखा हुआ पड़ा है. और इनके कारण इनकी किडनी है वो फेल हो जाती है, मसल्स है वो लूज हो जाती है, फर्टिलिटी पावर है कम हो जाती है, अंडों का जो सेल जो कवच होता है वो भी बहुत वीकर हो जाता है, अंडा गिरते ही टूट जाता है. सो दिस इज़ द स्टोरी ऑफ दिस इजिप्टियन वल्चर. और अब IUCN ने इसको एंडेंजर्ड स्पीसीज डिक्लेयर कर रखा है, लेकिन इस एरिया में इनका ब्रीडिंग रेट बहुत ज़्यादा है. हालांकि जो लंबे गिद्ध वाली जो है लॉन्ग बिल्ड वल्चर है, वाइट रम्प्ड वल्चर है, वो यहाँ पर नहीं है. लॉन्ग बिल्ड कभी कभी दिख जाता है, किंग वल्चर कभी कभी दिखता है, लेकिन यहाँ 100 किलोमीटर दूर तालछा पर ब्लैक बक सेंचुरी है, वहाँ पर वल्चर की 7 स्पीसीज आप नवंबर से फरवरी तक आप वहाँ पर आप देख सकते हो. और बीकानेर में एक जॉर्बेर एरिया है जहाँ पर पूरे सिटी की जो डेड जो गायें हैं कैटल से वहाँ पर आती है, और जॉर्बेर में इनकी संख्या, सारे वल्चर्स की संख्या करीब 2000 के आसपास आप वहाँ पर देख सकते हो. तो इस एरिया मेन जो वल्चर की जो साइट है वो बीकानेर का जॉर्बेर एरिया है. और अब इसमें रीजन क्लियर कट इंडिकेशंस नहीं है. साइंटिस्ट कहते हैं कि डाइक्लोफेनाक रेस्पॉन्सिबल है, कुछ कहते हैं पेस्टिसाइड रिस्पॉन्सिबल है, कुछ कहते हैं भी इनका हैबिटेट डिस्ट्रॉय हो गया, प्लांटेशन ख़त्म हो गया है, और कुछ ये है कि वायरस, नील ब्लू वायरस वो इसके लिए रेस्पॉन्सिबल बता रहे हैं. लेकिन क्लियर कट रीजन क्या है इनकी पॉपुलेशन डिक्लाइन के, वो अभी तक भी क्लियर नहीं है, क्योंकि 2 पैरेलल प्रोजेक्ट इंडिया में भी चला और नेपाल के अंदर भी चला, फंडेड बाई लेप्टर रिसर्च फाउंडेशन और पेरेग्राइन फंड, और ऑलमोस्ट सिमिलर ऑब्जर्वेशंस. लेकिन हकीकत यह है कि डाइक्लोफेनाक को बैन कर दिया, मेलॉक्सिकम मेडिसिन है वो चालू कर दी, लेकिन उसके वही साइड इफ़ेक्ट है जो डाइक्लोफेनाक के थे, क्योंकि ये सारा फंडेड प्रोजेक्ट है. मिनिस्टर में जो फॉरेस्ट मिनिस्टर है, वह वन मंत्रालय है, पैसे दे के जो फार्मा कंपनियां हैं वो अपने पेटेंट करवाती है, मॉलिक्यूल्स को रिलीज़ करती है, लेकिन एक्चुअल हकीकत क्या है वो भी अभी क्लियर नहीं है कि इस पॉपुलेशन डिक्लाइन के वास्तविक रीजन्स क्या है. और मेरे गांव में भी लॉन्ग जीपस जो बेंगालेंसिस है उसके 3 और 4 नीम के ऊपर ब्रीडिंग थी 92 में, मुझे याद है, लेकिन इन दिनों कोई ब्रीडिंग नहीं हो रही यहाँ पर. किंग वल्चर जो रेड चोंच वाला जो है वो भी कभी कभी दिख जाता है, लेकिन फिर भी इजिप्टियन वल्चर है ये एक सक्सेसफुल होप है डेड बाय बॉडीज़ को, है ना, सफाई प्रहरी के रूप में, क्लीनिंग एजेंट्स के रूप में. तो इस बर्ड्स में भी बहुत प्रेशर है डेड बॉडीज़ को. और इसके अलावा परियाकाइट है, वो एक कैरकस फीडर है यहाँ पर, रेवन है, ब्लैक डोंगो है, कैटल एग्रेट्स हैं. तो यहाँ पर 10-12 बर्ड्स हैं जो डेड बॉडीज़ को वो क्लियर करती है, स्कैवेंजर बर्ड्स हैं. लेकिन जो रोल लॉन्ग बिल्ड वल्चर का, वाइट रम्प्ड वल्चर्स का था वो नहीं है, क्योंकि कई बार देखा है पूरी जो कैटल्स है, गाय हैं, भैंस है, 1 घंटे में लॉन्ग बिल्ड वल्चर 1 घंटे में साफ़ ही कर देते पूरे मीट को, 2 क्विंटल की एक कैटल्स को। और अब आप सड़कों में जाते हैं, देखा होगा डेड बॉडी सड़ती रहती है, कितनी स्मेल। तो ये एक इसके अंदर भी एक चैलेंज है, यूँ मान लो। और लेकिन इस पर अपन क्या कर सकते हैं? ब्रीडिंग सेंटर है, ठीक है, पिंजौर के अंदर ब्रीडिंग सेंटर है, हिमाचल में भी है ना, BNHS है, वो भी इंक्यूबेशन सेंटर है, वो हैंडल कर रही है। लेकिन लेबोरेटरी में कितने कितने भाई हम एग को इंक्यूबेट करके रिलीज़ करेंगे। तो ये मेरा प्रेजेंटेशन था और ये हमारे स्टूडेंट का ही वर्क है। अभी भी इसमें चल रहा है, वर्क उनका चल रहा है, PhD थीसिस सबमिट होने वाली है। और फ्यूचर में हम इसमें कुछ एस्पेक्ट और लेंगे। एक तो कंपेरेटिव स्टडी हम करेंगे कि रूरल एरिया और जो अर्बन एरिया है उसमें जो इजिप्शियन वल्चर की ब्रीडिंग है उसमें क्या क्या डिफरेंस है, ये एक हमारा पैरामीटर रहेगा। और एक नेपोटिस्टिक बिहेवियर का, क्यों मेरे गांव में मैंने देखा है कि केडिया हवेली में ब्रीडिंग थी, उसके 100 मीटर दूर चांदगोटिया हवेली, उसमें ब्रीडिंग थी। तो मैंने देखा जो मेल जो पेरेंट था वो केडिया हवेली में भी फीडिंग करवा रहे हैं अपने पार्टनर को, और चांदगोटिया हवेली में जाके भी वो फीडिंग करवा रहे हैं। तो हो सकता है कि 1 साल उसकी पार्टनर वो रही होगी और नेक्स्ट ईयर वो पार्टनर चेंज होगी, लेकिन मेल सेम था। लास्ट ईयर पार्टनर दूसरी थी, इस साल पार्टनर दूसरी है। तो ये हेल्पिंग बिहेवियर है एक तरह का, नेपोटिस्टिक बिहेवियर इसको बोलते हैं। तो वो भी इसके अंदर स्टडी हमें करना है। और पेस्टिसाइड और बायोसाइड का इफ़ेक्ट क्या है वो हमें ऑब्जर्व करना है। एनवायरमेंटल स्ट्रेस का इफ़ेक्ट क्या रहेगा वो भी इसमें हमें ऑब्जर्व करना है। और आर्टिफिशियल नेस्ट इम्प्लांटेशन का भी हम सोच रहे हैं कि खेतों में आर्टिफिशियल नेस्ट इम्प्लांट करें तो क्या ये उसको यूज़ करेंगे। और एक सेक्स डिटर्मिनेशन का भी हमारा प्लान है, क्योंकि इसके फैदर में जो विंग बर्ड है उससे सैंपल लेके कि ये जो बच्चा डेवलप हो रहा है मेल है या फीमेल है, तो कैरियोटाइप इसमें डेवलप किया जाएगा और फ्लो साइटोमेट्री के द्वारा भी ये पैरामीटर है वो एनालाइज हो सकते हैं। तो इस बर्ड्स में अभी भी काफ़ी एस्पेक्ट है, इसमें भी 3-4 PhD की अभी गुंजाइश है और ये हम अभी लेंगे आने वाले दिनों में। सो इट इज़ ऑल अबाउट माय वर्क। और ये हमारे कुछ इसमें हेल्पर्स हैं, जे निग्रो, जे डोनाजार स्पेन से हैं, बिल्ड स्टेन ये हाक माउंटेन सेंचुरी पेनिसिल्वेनिया से हैं, मार्क एंडरसन ये फिश बैटिंग इंस्टीट्यूट साउथ अफ्रीका से हैं, VC सोनी गुजरात से हैं, रिटायर्ड हो गए अभी है, लेकिन वाइल्ड लाइफ में उनको एक्सपर्टाइज के रूप में अपॉइंट कर रखा है। और AN शर्मा ये मेरे PhD गाइड हैं, और प्रोफेसर हिलस्ट्रॉम ये भी USA से हैं। और पैराग्राइन फंड ने हमें सपोर्ट किया है इस प्रोजेक्ट को। एनी क्वेश्चन कोई? Our students are doing on black ibis, Egyptian vulture, Paria kite, black wing kite, black drongo, and one student is doing work on ethno-ornithology, that is birds and human relations. तो यहाँ के जो लोकगीत, पशु पक्षियों के लोकगीत है उनका वो कंपाइलेशन किया है उन्होंने 500 गीतों का, इंग्लिश में ट्रांसलेशन चल रही है। और यहाँ पर लोकोक्तियाँ हैं, गांव में आप जाओ बूढ़े आदमियों से मिलो तो फोकलोर्स बहुत है, तो करीब 2000 फोकलोर्स का कलेक्शन किया है जो एनिमल्स रिलेटेड है। जैसे एक रेमन यहाँ पर डॉट का एक पक्षी है, कौवे की वैरायटी, तो 250 ग्राम भोजन है वो 5 खुराक में खा जाता है। तो यहाँ पर एक लोकोक्ति है कि कागले की चाँच पाव की 5 पाव मतलब 250 ग्राम, तो 5 फीडिंग में वो 250 ग्राम फूड है वो खा जाता है। तो इस तरह की मतलब करीब 2000 के आसपास फोकलोर्स का कलेक्शन किया है। और गांव में रीति रिवाज है, टोने टोटके हैं जिसमें पशु पक्षियों का यूज होता है, जादू टोने यूज होता है। तो इस तरह का एक वर्क है इथनोलॉजिकल एस्पेक्ट में, वो हमारी एक स्टूडेंट PhD कर रही है इस टॉपिक के ऊपर।
Thank you, Dr. Saab. It is a really very good presentation and very informative, at least for me. Though I am not a concern having with this details about this geology of this world, but I am having great interest in that wildlife.
Okay.
Even I was having different kind of pets like reptile, birds, animals. So I am a very lover to this, and I was having pet of that part is even cobra.
Cobra, okay.
During my even after job, people surprised that one can have a pet of cobra. So that is very informative for me. And one thing I would like to supplement that one veterinarian is also working under me. So the cause of death and the chemicals that he's having great information about it. He's a very grassroots level worker. So I will communicate and discuss. And the question is that is there any other economic importance of these words besides the scavenging? It is used as a scavenger.
Yeah.
सफाई करने का काम आता है. इसके अलावा भी कोई इसका इकोनॉमिक
It
इम्पोर्टेंस है?
feeds on rats, gerbils, चूहों को ऊदबिलाव जो होते हैं, और खेतों के जो बड़े जो चूहे हैं
Pest
उनको
control.
उनको ये खाता है, तो क्रॉप प्रोटेक्शन में इनका रोल है.
That is very good.
बाकी तो स्कैवेंजर बर्ड्स है मेनली, तो बाकी ये गिलरी जो है स्क्विरल्स हैं, हैजहोग है.
और जैसे आपने बताया कि इनके बारे में कुछ किस्से कहानियाँ हैं. Likewise, I heard one story in my childhood that one herb that is chitrak I have mentioned in my presentation, that is Plumbago jalanica, and it is a very high medicinal value.
Okay.
So if somebody want to get a right stick of a Plumbago jalanica, then they have to have the nest of this yellow beak vulture.
Okay.
And how they can find that stick? They tied the legs of these जो जिसके बच्चे होते हैं ना चूजे, उनका chickens, and after some time they will find they are tying with the wires. After some time they will find that that chicken is released from that wire, because of they are bringing that stick, and that stick having power to melt down this one.
Okay.
तो इस तरह की मैंने कहानी सुनी थी कि उससे अगर आपको रियल चिताव और चित्र का लकड़ी चाहिए तो वैद्य लोग ऐसे कलेक्ट करते. तो इसके बारे में कुछ आपको कोई इन्फॉर्मेशन हो, कभी कुछ सुना हो?
हमारे यहाँ पर जो ट्राइबल जो एरियाज है, नोमेडिक जो पीपुल है, जैसे सांसी हैं यहाँ पर, आप खेतों में जैसे बीड में जाओ सालासर की तरफ, तो सांसी कम्युनिटी यहाँ पर है, कालबेलिया यहाँ पर है, साटिया कम्युनिटी यहाँ पर है, और इसके अलावा जो गांव के जो बुजुर्ग जो लोग हैं, पुराने जो मतलब जो असली जो रूरल लाइफ से जुड़े हैं, जिनका जीवन ही खेतों में बीतता है, तो उनके पास असली इंडिजिनस नॉलेज उनके पास है.
आई थिंक सम
बाकी
इथनोलॉजिकल
यंग जनरेशन है तो उनको तो भाई नाम का ही पता नहीं है. इवन तो मारवाड़ी भी नहीं आती है, मारवाड़ी गीत नहीं आते हैं, है ना. वो तो ठीक है,
लोकल लैंग्वेज में इसको जो एक तो काली वाली चील और पीली वाली चील,
इसको धोली चील बोलते हैं यहाँ पर, धोलती चील, वाइट कलर है.
तो दैट इज
और बर्ड्स में ये है कि अडल्ट यदि वाइट कलर का है तो उसके जो बच्चे होंगे वो ब्लैक कलर के होंगे. अडल्ट यदि ब्लैक कलर का है तो उसके बच्चे वाइट कलर के होंगे. और टाइम के साथ साथ उनका प्लूमेज कलरेशन है वो चेंज होता रहता है. और ये शायद थर्मो रेगुलेशन का एस्पेक्ट है. वाइट कलर यदि फैदर है तो वो मतलब बॉडी उनकी ठंडी रहेगी. इसके इमेच्योर आप देख रहे हो ब्लैक कलर के, तो उसको हीट ज़्यादा चाहिए, हीट ज़्यादा चाहिए, तो इनके फैदर्स हैं ब्लैक कलर के.
just like the cubs of that like lion, and they are having just like that natural habitat, according to natural habitat they are having that color, color is changing. जैसे
और ये बर्ड्स क्या है, गाय का गोबर भी खाता है.
अच्छा.
तो गाय के गोबर में क्या है, कैरोटीन है, और कैरोटीन के कारण इसकी चोंच का जो पीला रंग है वो डेवलप होता है. एक कैटल एग्रेट है यहाँ पर, वो भी गाय का गोबर खाता है, और ब्रीडिंग सीजन में मेल का पूरा चेस्ट, पूरी गर्दन एकदम सुनहरी रंग की हो जाती है. तो ये कैरोटीन की वजह से है. और डंग बीटल्स खाते हैं. डंग बीटल्स की जो इंसेक्ट की जो स्किन है उसमें काइटिन है. काइटिन है वो भी इसके पिगमेंटेशन के लिए रेस्पॉन्सिबल है.
So my supplement is that I think some ethnological work should be done. That is there any that kind of information we have to collect? That how it can help in searching that kind of, because you are having that during nesting that you have listed a lot of herbs and tree and some indicators like red cloth. So some ethnological studies should be done, and some literature that how they earlier they were doing that practice. So that will be, I think.
तो बर्ड्स में क्या है कि किचन और बेडरूम दो चीज़ चाहिए. आदमी को वही चाहिए भाई, रहने के लिए, सोने के लिए बढ़िया मचान मिल जाए, पलंग मिल जाए, खाने के लिए बढ़िया भोजन मिल जाए। तो कोई भी बर्ड्स हाउस में बेडरूम और किचन, दो चीज़ें यदि बर्ड्स को अवेलेबल है तो ब्रीडिंग रेट बहुत अच्छी होती है। और बेडरूम का मतलब जहाँ पर बर्ड्स नाइट को रेस्ट करता है, और किचन का मतलब जहाँ उसको फीडिंग मटेरियल कोई भी मिल जाए। और हर बर्ड्स की फीडिंग हैबिट अलग अलग है, कोई हर्बीवोरस है, कोई फ्रूगीवोरस है, कोई कार्निवोरस है, कोई स्कैवेंजर्स है। तो इनके फ्लाइट पाथवेज भी अलग अलग है, दिन में कहाँ किधर जाते हैं, शाम को किस डायरेक्शन से आते हैं। नेस्ट के भी फ्लाइट पाथवेज हैं। तो अवेलेबल ट्री कौन सा है, उसमें प्रीफर्ड ट्री कौन सा है, कैनोपी एरिया कितना है, हाइट कितनी है, वहाँ पर आसपास में प्रीडेटर्स तो नहीं है। तो नेस्टिंग सक्सेस है। लेकिन हमारे इलाके में, मेरे गांव में आसपास का देखता हूँ मैं, हर साल 50 बर्ड्स में ब्रीडिंग चलती है सक्सेसफुल, और 50 में करीब 30-35 बर्ड्स की हर साल इनकी पॉपुलेशन बढ़ रही है।
थैंक यू वेरी मच।
और यहाँ भी तौदी कॉलेज के सामने भी फॉरेस्ट एरिया है, वहाँ पर भी मैं कई बार फोटोग्राफी के लिए आता हूँ। सालासर एरिया है, इधर भी इस बर्ड्स की काफ़ी अच्छी ब्रीडिंग है।
Thank you, Professor,
Thank
for
you, sir.
your nice lecture. I want to ask that you have told that diclofenac sodium is one drug that is responsible for the death of these type of the vultures. So I have seen, you have said that this diclofenac sodium is still, it is banned in here, but I have seen they
बैन है, लेकिन गांव में यूज होती है, अभी भी यूज होती
it
है
is
चोरी
it,
छिपे।
it is being distributed through hospitals also in combination as well as the solo drug. So what would you suggest that whenever these type of the drugs, they are being expired, they are being dumped in the environment as such, and these vultures might be eating, taking that type of the drug, and that will be killing these type of the vultures. So can you suggest some type of the remedy that whenever these type of the drugs are being disposed of, they should not be taken by the animals, vultures?
अब वो तो फार्मर्स को ट्रेंड किया जाए, लेकिन मैंने देखा है वेटरी दुकानें हैं, वहाँ पर ये डाइक्लोफेनाक खुले में बिक रही है। ठीक है, उन्होंने स्टिकर लगा रखा है डाइक्लोफेनाक इज़ नॉट सोल्ड हियर, लेकिन रियलिटी है कि डाइक्लोफेनाक सोडियम है वो सेल होती है। और उसके अल्टरनेट में मेलॉक्सिकैम है, वो मार्केट में है, लेकिन मेलॉक्सिकैम भी सेम इफेक्ट है। क्योंकि सो मेलॉक्सिकैम बनाने वाली कंपनियों ने मिनिस्टर को पैसे दे दिए होंगे 200-400 करोड़ रुपये, वो क्योंकि सारा बिजनेस बनिया है सब, लेकिन रियलिटी है वो तो रियलिटी है। और जोधपुर में मेहरानगढ़ फोर्ट है, अब जोधपुर का कोई भी वहाँ पर कोई प्लांटेशन नहीं है, लेकिन मेहरानगढ़ फोर्ट में 300 लंबी गर्दन वाले जो वल्चर है, लॉन्ग बिल्ड वल्चर, उनकी सक्सेसफुल ब्रीडिंग हो रही है मेहरानगढ़ में। तो वहाँ के साइंटिस्ट तो ये कहते हैं कि भाई हैबिटाट लॉस का कोई इस पर इफेक्ट नहीं है, डाइक्लोफेनाक का कोई इफेक्ट नहीं है। तो आप कभी जोधपुर जाओ, मेहरानगढ़ की दीवार में आप देखोगे 300 लॉन्ग बिल्ड वल्चर की सक्सेसफुल ब्रीडिंग वहाँ पर है। तो जस्ट अपोजिट। फिर भी रियलिटी क्या है भाई, हम कह नहीं सकते, क्योंकि इतनी हमारे पास हाई फाई लेबोरेटरीज भी नहीं है, सिंपल सरकारी कॉलेज है, वहाँ पर चौक से हम पढ़ाते हैं, डस्टर से पढ़ाते हैं। और इवन तो हमारे पास तो इस तरह का भी प्रोजेक्टर भी नहीं है, इस तरह का ऑडिटोरियम भी नहीं है। फिर भी लिटिल सुविधाओं में हम वर्क कर रहे हैं, तो हमें संतुष्टि है।
Any other question?
What is much of you?
What inspired you to study about vulture?
What is?
Inspired.
प्रेरणा कैसे मिली? मैं मूलतः जंगली आदमी हूँ, गांव का रहने वाला हूँ, एकदम छोटा सा गांव है। और यूँ मान लो मेरे पूरे दिन के 15 घंटे खेतों में ही बीतते हैं। तो मैंने एमएससी चूरू से किया, लोहिया कॉलेज से, और वहाँ पर अनफॉर्चुनेटली ऑर्निथोलॉजिस्ट थे मेरे जो पीएचडी गाइड हैं, डॉक्टर एन शर्मा। तो वो एमएससी में जो थे तब 8 ही सीटें थी हमारे पास उस समय, अब तो 40 सीटें हैं। तो सभी बच्चों को खेतों में ले जाते हो हर संडे को हमारा मॉर्निंग 3-4 घंटे खेतों में ही। और मैं एक बढ़िया पेंटर भी हूँ, तो यहाँ के जितनी भी बर्ड्स की जो पेंटिंग है वो मैंने बनाई, वाटर कलर से पेंटिंग बनाई, उनका पूरा डिस्क्रिप्शन लिखा है, इसकी हैबिट कैसी है, बॉडी वेट कैसा है, ब्रीडिंग सीजन कौन सा है। और हर मंथ हम स्कूलों में जाके उनके एग्जीबिशंस भी लगाते हैं। तो मेरी बर्ड वाचर की जो हैबिटाट है वो 1989 से जैसे स्टार्ट हुई, और जॉब में आने के बाद में फिर यूजीसी के प्रोजेक्ट हम ले लिए छोटे छोटे, कि उसमें कैमरे भी नहीं होते थे, रील वाले कैमरे होते थे और बड़े जूम लेंस तो थे ही नहीं। चूरू में एक फोटोग्राफ के पास था 200 mm का जूम लेंस, और वो भी 20 रुपये घंटे का किराया लेता। तो फोटोग्राफी बहुत टिपिकल टास्क था। तो यूजीसी के पहले प्रोजेक्ट में हमने निकॉन का FM10 कैमरा ख़रीदा, दूसरे प्रोजेक्ट में एक बढ़िया बाइनोकुलर खरीदी, तीसरे प्रोजेक्ट में हमने सिग्मा का लेंस खरीदा 400 mm का, और अब जो ये कैमरा है मेरे पास इसमें 2000 mm का जूम लेंस है। उड़ती हुई हवाई जहाज़ की फ़ोटो भी मैं इससे खींचता हूँ, और इस कैमरे से मैंने 10 लाख फ़ोटो अब तक खींच ली है। लेकिन पीएचडी के दौरान मैंने 20,000 रील वाली जो फिल्म है वो खींची मैंने, 20,000 शॉट लिए, और उसमें 100 बेस्ट पिक्चर है वो मैंने मेरे थीसिस में लगाई। तो मेरे जो असली जो मोटिवेशनल है वो मेरे गाइड हैं डॉक्टर EN शर्मा। और सेंचुरी में विजिट करते, भरतपुर भी हम जाते हैं, तो वहाँ पर एक ये जो गाइड है रणधीरा नाम है उसका, पूरा इलिटरेट है, पढ़ा हुआ नहीं है बिल्कुल भी, लेकिन उन्होंने सालिम अली के साथ 20 साल काम किया है, और भरतपुर की हर बर्ड्स की स्पीसीज़ का जोलॉजिकल नेम उसको याद है, हर फिश का याद है, वहाँ पर कैंपस में जो फ्लोरा है उन सब के साइंटिफिक नेम याद है। और उस रणधीरा का जो नॉलेज है वो एक मतलब हमसे बहुत ज़्यादा फार बेटर है। तो आप कभी भरतपुर आपका जाना हो तो आप उनसे मिलना, कितना नॉलेज है। तो अभी 2 साल पहले की बात है, वहाँ पर ये पेलिकन बर्ड्स नहीं आए घाना सेंचुरी में, तो मैंने पूछा भैया हमें पेलिकन तो दिखाओ, है ना, पेलिकन्स नहीं है। तो उन्होंने बोला कि यहाँ पर पहले रिवर का पानी आता था, और रिवर के पानी के साथ मछलियाँ भी आती थी, और अब रियलिटी के यहाँ पर कुएँ की सप्लाई है, ट्यूबवेल है, उसका पानी आ रहा है, अंडरग्राउंड पानी है वो सप्लाई हो रहा है, उस पानी में मछलियाँ नहीं है, मछलियाँ नहीं है तो पेलिकन्स भी यहाँ नहीं है। और वो अजान बांध का पानी था वो रोक दिया गया भरतपुर में आने से, और सेंचुरी से 20 किलोमीटर दूर लोकल छोटे छोटे पॉन्ड बन गए, वहाँ पर पेलिकन्स थे। तो मतलब कितनी ऑब्जर्वेशंस आप उस रणधीरा की आप देखो, कितना मतलब नॉलेज है उसको। और कोई क्वेश्चन? हाँ हाँ हाँ, यूरोपियन स्पीसीज।
तो वो डू दे फ्लाई बैक टू देयर कंट्री?
हाँ,
और वो बच्चों को साथ लेके जाते होंगे?
हाँ नहीं, वो अडल्ट ही आते हैं, बच्चे उनके मैंने नहीं देखे।
बट व्हाइल दे विल कम बैक नेक्स्ट ईयर, बाय द टाइम चिल्ड्रन
नहीं, वो यहाँ पर केवल 4 महीने यहाँ रहते हैं, नवंबर से फरवरी, उसके बाद में चले जाते हैं। और यहाँ पर वो ब्रीडिंग भी नहीं करते हैं। यहाँ के जो इंडियन स्पीसीज उनके साथ भी उनका कॉर्टशिप नहीं होता है, कॉपुलेशन नहीं होता है।
सर, एक जगह आपने बताया कि वन मेल वल्चर,
हाँ,
whom you identified कि ये मेल है जो who was feeding 2 females।
हाँ, मेल में तो क्या है, वो बाइनोकुलर से साइज से पता लग जाता है। मेल जो है वो जनरली बॉडी उसकी हैवी होगी, बॉडी साइज मतलब बड़ी होती है, और थोड़ा एग्रेसिव भी होता है ब्रीडिंग सीजन के दौरान। तो ये मैंने देखा है, एक हवेली में फीमेल को फीड कर रहा है, फिर कैराकास्ट का टुकड़ा ला के उस नेस्ट में भी जाके फीड कर रहा है। तो उसका पिछले साल अफेयर रहा होगा उस फीमेल के साथ, इस साल अफेयर उसका दूसरी फीमेल से है। तो मेल होते हैं वो वैसे वफादार नहीं होते हैं, दे आर प्रोमिस्कुअस। कोई भी मेल हो, चाहे आदमी हो चाहे बर्ड्स हो, लेकिन ये मैंने देखा, ये नेपोटिस्टिक बिहेवियर है, हेल्पिंग बिहेवियर, ये अभी एक्सप्लोर करना है। इसमें टैगिंग लगा के, रिबन लगा के टांगों में रिंग करके, रेड कलर की, ब्लू कलर की, हम इस ऑब्जर्वेशन को प्रूव कर सकते हैं।
सर, एक ये भी कहा जाता है कि जैसे इन बर्ड्स के एग को किसी ने देख लिया या टच कर लिया तो उसमें वो फिर हैचिंग और
हाँ, ये भी हमने कोई, मैंने ब्लैक आइवी से वर्क किया, एक पीपल के नेस्ट पे जैसे 3 अंडे थे, और हमारा एक स्टूडेंट उसको मैं चढ़ा था कैमरा लेके, वो फोटोग्राफी करता, तो दूसरे दिन जाके देखा तो वो तीनों अंडे ही डिस्ट्रॉय हो गए। तो सैंपल ठीक है भाई, 50 सैंपल हम हाथ में रखते हैं, लेकिन फोटोग्राफी के लिए 3 और 4 सैंपल ही रखते हैं, क्योंकि हर नेस्ट की फोटोग्राफी करें तो फिर इस तरह की संभावना भी होती है। तो ठीक है, फोटोग्राफी हमने ले ली, अब तो ड्रोन कैमरे की सुविधा है तो ऊपर चढ़ने की जरूरत भी नहीं है, लेकिन ये होता है।
हाँ सर, ये कौवे कैसे डिसअपियर हो गए यहाँ इस इलाके से?
कौवों का भी वही कहानी है सर, मेन तो पेस्टिसाइड्स का यूज किया, इंसेक्टिसाइड, पेस्टिसाइड्स। और इवन तो यहाँ पर रेवन जो डॉट काग है, उसके फतेहपुर में अमृत आश्रम है, उसमें पाँच छः पैर हैं उसके, लेकिन डोडका का नेस्ट हमने आज तक नहीं देखा। 30 साल से जंगलों में घूम रहे हैं, डोडका दिखते हैं लेकिन उनका नेस्ट आज तक नहीं मिला, कहाँ वो ब्रीड करते हैं। और हाउस क्रो की भी पॉपुलेशन बहुत कम हो गई है। और हमारा एक स्टूडेंट है हेमंत सोनी, वो हर साल 1000 आर्टिफिशियल नेस्ट है बना के डोनेट भी करता है, खाती से बनवा के। तो घरों में अभी स्पैरो डे आएगा, उससे हम वो डोनेशन हम स्टार्ट कर देंगे। तो पिछले 3 साल में 3000 नेस्ट हमने पूरे चूरू सिटी में इम्प्लांट किए हैं, ग्रेवयार्ड के अंदर, स्पोर्ट्स कॉम्प्लेक्स में, स्कूलों में, मंदिरों में, हर घर के अंदर।
सर, ये जैसे प्रोफेसर अश्वथ वाज आस्किंग यू कि डू दीज बर्ड्स हैव एनी अदर प्रोग्राम एजेंडा मोर देन स्कैवेंजिंग, तो कभी ये भी लगता है ना जैसे डंग को खाता है, उसमें कुछ देर आर सीड्स, तो वाइल फ्लाइंग बैक एक सीड इस जगह का दूसरी जगह,
हाँ बिल्कुल, पॉलिनेशन,
सो दैट हर्ब इज आल्सो स्प्रेड ओवर।
हाँ, डिस्पर्जल में भी इसका रोल
है ना।
और ये तरबूज भी खाता है, मतीरे खाता है, काकड़िए खाता है, मतलब प्योरली कार्निवोरस नहीं है। चपातियाँ भी खा रहा है, मिठाई भी खा रहा है, गुलाब जामुन खा रहा है, चमचम खा रहा है। तो मैं देखा है अलग अलग तरह की हैबिटाट है। और श्राद्ध पक्ष से हम स्वीट तो ले जाते हैं कौवों को परोसने, कागोस बोल के, और इजबिटल वल्चर आ जाते हैं, वो खा के चले जाते हैं। तो मेरे को वार्निंग मिली है साउथ अफ्रीका से कि यू शुड गो टू जेल, यू आर चेंजिंग फीडिंग हैबिट ऑफ दिस बर्ड। और हमारा पेपर वो पब्लिश ही नहीं हुआ, ठीक है, न्यूज़पेपर में छपा पूरे हेडिंग पूरे राजस्थान में, और वल्चर भी खाने लगे हैं लजीज व्यंजन। तो फीडिंग हैबिट चेंज हो रही है। हम भी बर्गर खाते हैं, पिज़्ज़ा खाते हैं, हम भी देसी फूड खाते ही नहीं हैं।
सर, कुछ 2-4 जो यहाँ कहावतें कहते हैं ना कि आप बर्ड्स के बारे में हैं, तो कोई 2-4 और सुनाओ तो अच्छा लगेगा हमें।
देखता हूँ मेरे पास इसमें है तो पहले मेरा ये था, नवीन सर मैंने बोला था कि बर्ड्स इन फोकलोर फोक सॉन्ग, तो लोकगीत है 500, क्योंकि मेरी बच्ची इस पे पीएचडी कर रही है। फिर भी मैं देखता हूँ इसमें मोबाइल में भी स्क्रीनशॉट है तो मैं शेयर करता हूँ। कुछ कहावतें हैं जैसे कागलो हंस की चाल चाल तो आपकी ओर भूल गयो। कौवा मतलब हंस की चाल को मिमिक्री कर रहा है, तो हंस की चाल सीखते सीखते अपनी खुद की चाल और भूल गया वो। एक पिकॉक की है जैसे, मोरियो प्यूं प्यूं तो घणो ही करे, पण बरसनो तो इंदर के हाथ में। बाहर गालने से कोई रोटी नहीं गालेगा, प्यूं प्यूं करने से रेनफॉल नहीं होगी। एक और है, पूरी याद नहीं है, बेटी मरी जवाई चोर, इस तरह की सी कहावत है। तो जिस घर में बेटी मर जाती है उस जवाई का ससुराल में कोई रेस्पेक्ट नहीं होता है। बेटी मरी जवाई चोर। एक और कहावत है कि कुत्तो तो मरियो पण ठाकरा को ठा भी पड़ो। कुत्ता तो मर गया लेकिन ठाकुर का कुत्ता था भाई। बड़े जो रजवाड़े होते हैं इसमें तो वही है ना, यस सर है, जीव कम जीव कम। अब वो राज परिवार का कुत्ता बड़े है ना, टिकाई ठाकर का कुत्ता ले मरने के बाद में वो गली में फेंक दिया गया। तो कुत्तो तो मरियो पण ठाकरा को ठा भी पड़ो। ठा पड़ो मतलब पता चल गया।
सर, मैंने इसलिए पूछा आपसे क्योंकि मैं भी यहाँ लिटरेचर में करता हूँ काम।
हाँ।
तो स्पेनिश, इंग्लिश एंड राजस्थानी, जिसको मारवाड़ी कहते हैं,
हाँ,
इनकी कहावतों का कंपैरेटिव स्टडी एक हमारे दिमाग में है।
हाँ,
तो
मेरे पास पड़ी है सर लिखी।
हाँ, तो
करीब 2500 फोकलोर्स मेरे पास है।
तो
हर एनिमल्स की, कुत्ते की, गधे की, ऊँट की, मतलब एवरीथिंग। और पुराने लोकगीत हैं, पौधों के लोकगीत हैं, नीम के हैं, पीपल के हैं, बरगद के हैं, और पक्षियों के भी हैं। जैसे एक लाइन जैसे सुनाता हूँ मैं, ये जो गांव की औरतें जब दोपहर में होती है, बूढ़ी औरतें गाती रहती है वो, मारे हरिये वन का सुवटिया, तन राम मिले तो कहिए रे, तन राम मिले तो कहिए रे, तन श्याम मिले तो कहिए रे, मारे हरिये वन का सुवटिया, तन राम मिले तो कहिए रे। तो पुरानी औरतें गाती है कि तुम, मतलब सुवटिया मतलब तोता, पैरेट, तो उसको वो कह रही है कि भाई तुम्हें राम मिले, घनश्याम मिले तो मुझे भी बताना। इसमें देखता हूँ मैं मोबाइल में, यदि स्क्रीनशॉट है तो मैं 2-4 आपको शेयर करता हूँ। क्यों, मैं अभी मेरे गांव की एक किताब लिख रहा हूँ मैं, रतनगर की हिस्ट्री, रतनगर की एक विरासत टाइटल है उसका, और 6 साल से कंपाइलेशन चल रहा है, टाइप हो गई है और प्रूफ रीडिंग चल रही है। 3-4 महीने बाद उसको हम रिलीज करेंगे। हाँ, इसमें मिल गया स्क्रीनशॉट। पीकॉकी मैंने बता दी, मोरियो पीऊँ पीऊँ तो घणो ही करे, पण बरसनो तो इंदर के हाथ में। मोर बिना डूंगर किसा, यानी पीकॉक के बिना, डूंगर मतलब पहाड़, मोर बिना डूंगर किसा, मैं बिन किसी मलार, मैं मतलब बरसा, लुगाई बिना तीज किसी, बींद बिना किसो त्यौहार। तीज मनानी है तो लुगाई चाहिए, और त्यौहार मनाने तो बींद भी चाहिए। अकेले लेडीज से कोई भी फेस्टिवल है वो, कि हमारे एक पुराने सेठ थे, गोविंद जी अग्रवाल सेठ थे, वो इस तरह की बनाते थे खुद भी। तो उनका एक कोटेशन है कि लक्ष्मी जी को धिराणो घुघू, भाई पैसा तो सेठों के पास ही है, और घुघू मतलब उल्लू, यहाँ पर लोकल उल्लू है, बाण उल कोचरी जिसको बोलते हैं, उसको घुघू बोलते हैं। तो लक्ष्मी जी को धिराणो घुघू, जठे भी जावे, आपकी बिरादरी में जावे, आपकी घर आरी ने सागे ले जावे। तो सेठों में आपने देखा होगा कि कोई भी पाया भरना है, कोई प्लांटेशन करना है, कोई कॉलेज की बिल्डिंग बनानी है, अपनी वाइफ को वो साथ में रखते हैं। तो लक्ष्मी जी को धिराणो घुघू, जठे भी जावे, आपकी बिरादरी में जावे, आपकी घर आरी ने सागे ले जावे। लेकिन अब तो ये है कि भाई वाइफ पीर गई है तो चलो है, वी फील वेरी हैप्पी। तो कल्चर है वो चेंज हो रहा है। एक लोकोक्ति है, मेव और मोर ऊंची जगह ही राजी रहे। मोर है वो ऊंचे ठिकाने पे रहना पसंद करता है, और मेव है ये एक ट्राइबल वो है, नोमैडिक कम्युनिटी है जो घूमती रहती है, लेकिन जब ये जंगलों में रहेगी ऊंचे टीले के ऊपर अपना तंबू बांध के ऊपर वहाँ पर स्टे करेगी। तो मेव और मोर ऊंची जगह ही राजी रहे, मतलब हाइट वाली। बागा में दाख पाके, जब कागले का कागलिया फूल जा। अब ये फंगस पे जो फंगल वाले एस्पेक्ट है, तो अंगूर के पेड़ पे जब नेचुरली अंगूर जब पकता है, दाख में कन्वर्ट होती है, तो उसमें फंजाई भी डेवलप हो जाती है, और कौवे जब उस फंगस लगी दाख को खाते हैं तो कौवे के जो टॉन्सिल्स है उसके अंदर इन्फेक्शन हो जाता है। तो उसकी एक कहावत है कि बागा में दाग पाके जब कागले का कागलिया फूल जा, कागलिया मतलब टॉन्सिल। एक लोकोक्ति है, दाना चुगती मोडी मरी, मोडी मतलब अपने जो रिंग डव कॉमेडी जिसको बोलते हैं, दाना चुगती मोडी मरी, गादड़ मरियो तीसो, तीसो मतलब प्यास के बिना मर गया, अब के खोज ठाकना का निकला, जड़ मूल से जाशो। तो ये ठाकरों से रिलेटेड एक लोकोक्ति है। एक तीतर के लिए कहावत है, पाटराई जो होता है, तो तीतर में क्या है कि मेल पार्टनर है उसको थोड़ा कम सुनता है, ऊंचा सुनता है, बोला है जो मान लो डी फॉर डम्प। तो फीमेल तीतर अपने हस्बैंड को बोल रही है, तो वो कह रही है कि तीतर बोलो बोड़ा, मतलब अपने गांव में भी होता है, बूढ़े हस्बैंड है बूढ़े हो जाते हैं तो बोड़ा पटीर बोड़नर, तो तीतर बोले बोड़ा, और उसका वो मेल पार्टनर बोल दे अभी बेरों को नहीं किए तो 15 के 16। तीतर बोले बोड़ा कि अभी बेरों को नहीं किए तो 15 के 16। तीतर जाने तीतर को, मैं जानूं तेरे भीतर को। तो आपके मन में क्या है उसको आप या तो खुद जाने या आपका खुदा जाने, है ना। आपके मन में क्या है वो कोई भी नहीं जान सकता। टूटी डाली उड़ गया मोर, बेटी मरी जवाई चोर। तो बेटी मरने के बाद जवाई की ससुराल में कोई कदर नहीं होती है। एक ये फोकलर बहुत साइंटिफिक है, चीड़ी जे नावे धूल में, मतलब सैंड बात यदि करती है कोई हाउस स्पैरो, चीड़ी जे नावे धूल में आवन हार, यानी बरसात आने वाली है, पानी में नावे चिड़कली में विदा 3 बार। तो चिड़िया यदि मिट्टी में सैंड बात कर रही है इसका मतलब रेनफॉल आने वाला है, और चिड़िया यदि पानी के अंदर बात कर रही है इसका मतलब मानसून सीजन है वो जाने वाला है। चिड़े चिड़ीरी क्या की लड़ाई, तू चाल मैं आई। तो कई बार अपने हस्बैंड वाइफ नोक झोंक हो जाती है, लेकिन नाइट में कॉम्प्रोमाइज हो जाता है। तो चिड़े चिड़ीरी क्या की लड़ाई, तू चाल में आई। एक स्पैरो का नेस्टिंग का एक लोकोक्ति है, चिड़िया जे आड़ो करे, आड़ो मतलब ना घोंसला नेस्ट बना रही है, चिड़िया जे आड़ो करे कोठा कंगूरा माए। कोठा मतलब पुरानी जो हवेलियां हैं, कंगूरा मतलब टोडा। तो चिड़िया जे आड़ो करे कोठा कंगूरा माए, बिरखा आया आगमन तो 4 माह बरसाए। तो दी हाउस स्पैरो यदि कोठे के अंदर कंगूरे के अंदर ये अपना घोंसला बना रही है, इसका मतलब उस साल 3 और 4 महीने रेनफॉल रहेगा।
ठीक है सर, थैंक यू।
एक और है, कीड़ा पड़े गोबर कमाए। गोबर मतलब ये मक्खी के मैगट्स जो है, कीड़ा पड़े गोबर कमाए, पपी हो मीठो बोल सुहाए, अमल चामड़ी गिल्ली होए, बिरखा हुए न सांसों कोई। तो मतलब घरों में जो लेदर है, आपके जूते हैं, यदि लेदर यदि सॉफ्ट हो रहा है क्योंकि मौसम है और नम हो गया, ह्यूमिड हो गया, इसका मतलब रेनफॉल आने वाला है। तो इस तरह की काफ़ी है, 2000 से ज़्यादा कलेक्शन है हमारे पास, बर्ड्स का, एनिमल्स का, प्लांट्स का। और इस पर मेरी बच्ची PhD कर रही है, उसके बाद में इसको हम पब्लिश भी करेंगे।
PhD हिंदी लिटरेचर में है क्या?
ना, वो जूलॉजी की स्टूडेंट है।
अच्छा।
तो इथनो स्टडी ऑफ इथनो जोलॉजिकल एस्पेक्ट्स इन शेखावाटी रीजन ऑफ राजस्थान, ये उसका टाइटल है। और अजमेर यूनिवर्सिटी से वो PhD कर रही है।
इसको सर हम इंटरनेशनल लेवल पे भी करेंगे,
हाँ।
इंग्लिश और स्पेनिश और फ्रेंच, जर्मन, किसी साथी को जोड़ के,
हाँ।
और इसको थोड़ा और बड़ा लुक देंगे।
हाँ, नहीं हम कोलैबोरेटिव करेंगे
हाँ।
आपके साथ।
ठीक, थैंक यू सर, थैंक यू वेरी मच।
ओके सर।
Thank you so much, sir. Your generosity, spirit, and willingness to share your knowledge and expertise with us is truly reflective of the kind of great personality that you are. Thank you so much. Your presence is illuminating. Now, everyone, let us begin technical session 4, which is chaired by Dr. B.K. Goyal, Dr. A.K. Das, and Dr. Sarita Khaturia. Now, the session is open for oral presentations part 1, which is coordinated by Dr. Narendra Kumar. And at the same time, poster presentation and evaluation is about to start in ABB building, 2nd floor. Now I request Ms. Divya Yadav to take over from here.
afternoon everyone, this is Divya Yadav. I would like to invite the students for the oral presentation. And the first student is Divyanshi Sharma. Serial number is poster serial number 3.
Namaskar everyone, my name is Divyanshi Sharma. I am an undergraduate BSc student pursuing BSc in PCM. Okay, sir. So my topic is water conservation and I am here to spread awareness upon the topic water resource. So catch the rain where it falls, when it falls. It is a slogan given by Jal Shakti Abhiyan. जहाँ पे भी पानी गिरे, जब भी पानी गिरे, उसको वहीं पकड़ लो. So to start, मैं अपना इस अवेयरनेस सेशन को एक स्टोरी से स्टार्ट करूंगी. दिस इज़ अ स्टोरी ऑफ अ स्मॉल 10-year गर्ल नेम्ड लिली. लिली एक छोटी बच्ची है जो ग्रीनवेल टाउन में रहती है. इसकी अच्छी फैमिली है, हैप्पी फैमिली है. सो लिली का जो टाउन है वो ग्रीनवेल टाउन है एंड वहाँ पे अच्छी नदियाँ हैं, ग्रीनरीज हैं एंड काफ़ी बहुत प्यारा सा वो गांव है. वहाँ पे एक पार्क है जिसमें एक फाउंटेन था. तो लिली क्योंकि छोटी 10 साल की बच्ची है, बहुत चुलबुली सी, तो उसको ऐसे क्यूरियोसिटी हुई कि मैं जाऊंगी और इस फाउंटेन का पानी को टच करूंगी. जैसे ही उसने उस फाउंटेन के पानी को टच करा, वो एक कम्प्लीटली डिफरेंट वर्ल्ड में आ गई जहाँ पे लोगों को पानी के लिए बहुत स्ट्रगल करना पड़ रहा था. वहाँ पे कीचड़ के पानी से लोग कपड़े धो रहे थे, पानी पीने के लिए उनके पास पानी नहीं था, कुएँ से पानी निकालने के लिए उनको काफ़ी टाइम वेट करना पड़ता था. ड्रॉट की कंडीशंस बहुत कॉमन थी. इतना देख के वो बहुत घबरा गई. जैसे ही वो अपने ओरिजिनल वर्ल्ड में एंटर करी, उसने देखा कि एक गॉड ऑफ वाटर उसके सामने आया और उसने उसको बोला कि मैंने तुमको सिलेक्ट करा है ताकि तुम लोगों में अवेयरनेस फैला सको कि वाटर कितना इंपॉर्टेंट रिसोर्स है. एंड देन उसने जाके अपने फ्रेंड से बात करी कि मेरे साथ ये इंसिडेंट हुआ है और अपन को वाटर कंजर्व करना है, तो हम लोग उसके लिए क्या मेजर्स ले सकते हैं. देन लिली ने अपने वाटर कंजर्वेशन पे अवेयरनेस फैलानी शुरू करी. उसने बताया कि वाटर कंजर्वेशन एक ऐसा मेथड है जिसमें हम वाटर जहाँ पे भी है उसको हम कंजर्व करने का ट्राई करते हैं, चाहे वो अपनी अभी रिसेंट नीड्स के लिए एंड फ्यूचर नीड्स के लिए भी. फिर उसने 5 वाटर कंजर्वेशन टेक्निक्स बताई विच इंक्लूड्स वाटर कंजर्वेशन टिलेज, सॉइल मॉइस्चर कंजर्वेशन, ज़ेरोस्केपिंग, रेन वाटर हार्वेस्टिंग एंड ड्रिप इरिगेशन. नंबर 1 इज़ कंजर्वेशन टिलेज. इस मेथड में हम क्या करते हैं, दिस इज़ अ मेथड जिसमें हम सॉइल को भी कंजर्व कर सकते हैं, वाटर को भी कंजर्व कर सकते हैं. सॉइल का जो हेल्थ है वो भी इन्हांस होता है एंड वाटर भी कम यूज़ होता है. तो इस मेथड में क्रॉप्स के रेसिड्यूज़ हम साथ में रख देते हैं ताकि उनको न्यूट्रिएंट्स, मिनरल्स सारे मिलते रहें और सॉइल जो पानी एब्जॉर्व करता है वो भी कम हो जाए. तो दिस इज़ अ गुड मेथड. और क्योंकि वो ग्रीनवेल टाउन में रहती थी एंड वी ऑल नो दैट 70% ऑफ आवर वाटर इज़ यूज्ड इन एग्रीकल्चरल फील्ड, तो वहाँ पे सबसे ज़्यादा वाटर को कंजर्व करने की ज़रूरत है. नंबर 2 वाज सॉइल मॉइस्चर कंजर्वेशन. इसमें जितना मॉइस्चर को एब्जॉर्व करता है ह्यूमिडिटी को, उसको हम लोग कंजर्व करेंगे. दिस इज़ डन बाय क्रॉप रोटेशन. क्रॉप रोटेशन में क्या होता है, हम एक ही फील्ड पे डिफरेंट टाइप्स के क्रॉप ग्रो करते हैं. अगर हमने ईयर 1 में कॉर्न, ईयर 2 में भी कॉर्न, 3 में भी कॉर्न, 4 में भी कॉर्न ग्रो कर दिया, तो बहुत सारा पानी लगेगा एंड सॉइल भी इतना मतलब प्रॉपर प्रोडक्टिविटी नहीं देगा. वहीं अगर हम लोग ईयर 1 में कॉर्न, वीट, सोयाबीन एंड देन कोई फ्लावरिंग प्लांट ग्रो कर लें तो पानी भी कम लगेगा, सॉइल का हेल्थ भी अच्छी रहेगी. नंबर 3 इज ज़ेरस्केपिंग मेथड. इन दिस मेथड हम लोग उन पर्टिकुलर एरियाज जहाँ पे ड्रॉट की कंडीशंस बहुत फैमिलियर है उन एरियाज पे हम ड्रॉट रेजिस्टेंट प्लांट्स को ग्रो करना प्रेफर करते हैं. जैसे सम ड्रॉट रेजिस्टेंट प्लांट्स आर एलोवेरा, कैक्टस, फिलोडेंड्रन. तो इस टाइप के प्लांट्स को स्पेशली इन राजस्थान साइड एरियाज हम लोग इस ज़ेरस्केपिंग मेथड को ज़्यादा वो करते हैं, प्रेफर करते हैं. नंबर 4 इज रेन वाटर हार्वेस्टिंग. ये तो थोड़ी ऐसे कॉमन मेथड है. हम लोग क्या करते हैं, जो रूफ टॉप्स होते हैं उनको ऐसे स्लाइडिंग वे में डाल देते हैं सो दैट इट कैन बिल्ड अ कैचमेंट एरिया एंड देन वो रेन वाटर सारा एक टैंक में स्टोर हो जाता है विच कैन बी यूज्ड फॉर आवर डेली पर्पस. दिस इज रेन वाटर हार्वेस्टिंग. So a common type of rainwater harvesting which is seen in Rajasthan is the खादन system. यहाँ पे roofs को sliding way में नहीं बना के हम लोग जो land है उसमें ऐसे ढलान बना देते हैं with the help of mud which is commonly known as खादन and वहाँ पे सारा water store हो जाता है. Then next is drip irrigation. According to me, this is the very preferred and very accurate way to conserve water because यहाँ पे जैसे कि ग्रीनवेल टाउन पे या फिर वो एरियाज जहाँ पे एग्रीकल्चर ज़्यादा होते हैं वहाँ पे सबसे अच्छा मेथड है वाटर को कंजर्व करने का ड्रिप इरिगेशन. इन दिस मेथड जैसे अपन को पता है हम लोग पौधों पे पानी डालते हैं, काफ़ी पानी इधर उधर उछल के चला गया या पत्तों पे रह गया तो उस पानी को हम कैसे कंजर्व करें. दिस इज़ फ्रॉम ड्रिप इरिगेशन. इसमें हम पाइप्स लगा देते हैं. एज यू कैन सी हमने पाइप्स लगा दी एंड डायरेक्टली रूट्स को ही वाटर सप्लाई होगा सो दैट वाटर का वेस्टेज बहुत ज़्यादा कम हो जाए. दिस इज़ ड्रिप इरिगेशन. आई गेस दिस इज़ द बेस्ट मेथड टू कंजर्व वाटर. अब फिर वहाँ के लोगों ने क्वेश्चन पूछा कि चलो हमने वाटर तो कंजर्व कर लिया जैसे तैसे, क्या हम वो वाटर डायरेक्टली पी सकते हैं या डायरेक्टली हम किसी भी काम पे यूज़ कर सकते हैं, विच इज़ नॉट पॉसिबल क्योंकि उसमें बहुत सारी इम्प्योरिटीज़ होंगी. तो लिली ने बताया चलो वाटर कंजर्वेशन के बाद वाटर प्योरीफिकेशन के मेथड्स पे भी स्विच करते हैं. Then she explained water purification. So water purification is the method in which we remove all the contaminants, not all, approximate maximum number of contaminants from the water and such as bacteria, viruses or any organic material which is removed from water by the method of water purification. So 5 common water purification methods are by the use of hydrogen peroxide, UV treatment, reverse osmosis, ion exchange method and water chlorination. नंबर 1 इज़ हाइड्रोजन पेरोक्साइड. इसमें हम लोग जब वाटर में हाइड्रोजन पेरोक्साइड की बीड्स डालते हैं तो वो वाटर के कंटेमिनेंट्स को ऑक्सीडाइज कर देती है. द केमिकल रिएक्शन इज H2O2. इट विल बी कन्वर्टेड इनटू H2O एंड O2. ऑक्सीडाइज करती है इम्प्योरिटीज़ को और वो सारी इम्प्योरिटीज़ ऊपर आ जाती है विच कैन बी सेपरेटेड ईजिली. द नंबर 2 इज़ यूवी ट्रीटमेंट. हम लोग इसमें यूवी की बीम को मारते हैं वाटर पे ताकि जितने भी बैक्टीरियाज और वायरसेस वगैरह हैं वो सारे के उनके डीएनए पे जाके वो यूवी बीम लगती है एंड देन वो उनका रिप्रोडक्शन कम कर देता है एंड वाटर प्योरीफिकेशन की मतलब वाटर प्योरीफाई हो जाता है. नंबर 3 इज़ रिवर्स ऑस्मोसिस. इसमें हम लोग एक सेमी परमिएबल मेम्ब्रेन का यूज़ करते हैं जिसका साइज़ अप्रॉक्सीमेट 0.1 नैनोमीटर का होता है. ऑब्वियसली 100% वाटर प्योरीफाई नहीं हो सकता. The try is we can purify 90 to 92% of water. सेमी परमिएबल मेम्ब्रेन लगा दी एंड देन वाटर को हम प्रेशर करते हैं एंड वो सारा कंटेमिनेंट्स इज पास्ड फ्रॉम द सेमी परमिएबल मेम्ब्रेन. सारा फ्रेश वाटर निकल जाता है एंड कंटेमिनेंट्स एक साइड रह जाते हैं. नंबर 5 इज़ आयन एक्सचेंज मेथड. यू कैन सी आयन एक्सचेंज के ऐसे कैंपर्स बने हुए होते हैं जिसमें हम रेजन्स डालते हैं. रेजन्स कंटेन्स आयनाइज्ड these are ionized material जिसमें प्योर आयंस रहते हैं वो जाके इम्प्योर आयंस के साथ एक्सचेंज हो जाते हैं एंड देन उन इम्प्योर आयंस वाले रेजन्स को हम निकाल देते हैं एंड वो हो जाता है. यस यस. एंड वाटर क्लोरिनेशन, हम क्लोरीन डाल देते हैं वाटर में एंड विच इज़ हेल्प इन प्योरीफाइंग द वाटर. फिर इतनी बात सुन के वहाँ के लोगों को प्रैक्टिस हो गई कि हाँ हमको वाटर कंजर्वेशन कैसे करनी है और वहाँ पे कुछ लोगों ने वाटर कंजर्वेशन प्रैक्टिसेस स्टार्ट कर दी. कुछ गवर्नमेंट स्कीम्स हैं, जल शक्ति अभियान, कैच द रेन वेयर इट फॉल्स व्हेन इट फॉल्स एंड दीज आर सम 7 लिस्टेड स्कीम्स विच आर इनिशिएटेड बाय द गवर्नमेंट ऑफ इंडिया टू कंजर्व एंड प्योरीफाई वाटर. सो लिली आपका इतनी सारी चीज़ें सुनने के लिए बहुत ज़्यादा थैंक यू करती है एंड वो होप करती है कि आप लोग अपने वाटर कंजर्वेशन और वाटर प्योरिफिकेशन को डेली लाइफ में इम्प्लीमेंट करेंगे. थैंक यू. सर नहीं लिया सर, क्योंकि मुझे जो ज़्यादा प्रेफर्ड मेथड्स लगे मैंने उन्हीं का यूज़ किया. सर ऑस्मोसिस में हम लोग एक मेम्ब्रेन लगाते हैं एंड देन उससे मतलब इट इज़ अ सेपरेशन मेथड जिसमें हम फिर वो इम्प्योरिटीज़ एंड प्योर वाटर को सेपरेट करते हैं. सर रिवर्स ऑस्मोसिस में हमको प्रेशर अप्लाई करना पड़ता है, ऑस्मोसिस में नहीं. सर सॉल्यूशन is a mixture of solvent and solute. We solvent, because solute is a kind of impurities. It contains impurities. Thank you, sir. Thank you.
Thank you, Divyanshi. Next one is Sunidhi Garg.
Good morning, my name is Sunidhi Garg, and on today's national seminar, my topic is environmental chemistry, toxicology and health hazard, and also I am giving some solutions regarding this topic. This is my abstract. And what is environmental chemistry? First of all, it is a branch of chemistry which deals with the chemicals and other pollutants in the environment resulting directly and indirectly from human activities. इसका मतलब जो भी हम ह्यूमंस कर रहे हैं इन्वायरमेंट में वो डायरेक्टली और इनडायरेक्टली हमारे इन्वायरमेंट को इफ़ेक्ट करते हैं. एंड कंपोनेंट्स ऑफ इन्वायरमेंट, तो इन्वायरमेंट में मेन 3 कंपोनेंट्स होते हैं: नेचुरल कंपोनेंट्स, ह्यूमन मेड कंपोनेंट्स एंड ह्यूमंस. नेचुरल कंपोनेंट्स के अंदर एटमॉस्फेयर, लिथोस्फीयर, बायोस्फीयर एंड हाइड्रोस्फीयर आता है, जहाँ पे ह्यूमन मेड कंपोनेंट्स के अंदर बिल्डिंग, पार्क्स, रोड्स, मॉन्यूमेंट्स, ये सब चीज़ें आती हैं. एंड ह्यूमंस के अंदर इंडिविजुअल ह्यूमन या फैमिली या फिर कम्युनिटी, ये सब ह्यूमन कंपोनेंट्स के अंदर आते हैं. इसके अलावा जो हमारा एटमॉस्फेयर का एनवायरनमेंट है उसके अंदर एटमॉस्फेयर, हाइड्रोस्फीयर, एंथ्रोस्फीयर, लिथोस्फीयर, बायोस्फीयर एंड क्रायोस्फीयर, ये सभी स्फीयर्स मौजूद होते हैं. अब द एटमॉस्फेयर इज़ अ मिक्सचर ऑफ गैसेस दैट सराउंड द अर्थ. यहाँ पे जो हमारी एटमॉस्फेयर है इसके अंदर हमारे पास में मल्टीपल लेयर्स होती हैं. पहली लेयर है ट्रोपोस्फियर. ट्रोपोस्फियर वो होती है जिसमें हम सभी लिविंग बीइंग्स रहते हैं और यहाँ पे क्लाइमेट चेंज अकर होता है, मतलब क्लाइमेट चेंज होता है. इसके अलावा जो सेकंड लेयर है वो है स्ट्रेटोस्फियर, जो कि 12 किलोमीटर्स अबव होती है अर्थ से. इसके बाद में है मेसोस्फियर. मेसोस्फियर के अंदर जो है मीटियोरॉइड्स जो है बर्न हो जाते हैं, ये 50 किलोमीटर्स ऊपर होती है अर्थ से. इसके बाद में है थर्मोस्फियर जो 80 किलोमीटर्स ऊपर होती है अर्थ से. एंड लास्ट है एक्सोस्फियर जो 700 किलोमीटर्स टू 10,000 किलोमीटर्स होती है. ये सबसे लास्ट लेयर होती है हमारे एटमॉस्फेयर की. इसके बाद में है हाइड्रोस्फियर. हाइड्रोस्फियर इज़ द टोटल अमाउंट ऑफ वाटर ऑन अ प्लैनेट. मतलब हमारे अर्थ पे जितना भी वाटर है, फिर वो चाहे एयर में हो या अंडरग्राउंड हो या ओशियंस में हो, वो सभी हाइड्रोस्फियर के अंदर आते हैं. The third is lithosphere. Lithosphere is the solid outer part of earth. यहाँ पे लिथोस्फीयर के अंदर हमारे पास में होता है क्लस्टर्स, इसके बाद में मेटल्स, आउटर कोर और इनर कोर. इसके बाद में है हमारा एंथ्रोस्फीयर. द एंथ्रोस्फीयर encompass the total human presence throughout the earth system, including our culture, technology, built environment and associated activities. एंथ्रोस्फीयर के अंदर जितने भी हमारे कंपोनेंट जैसे बायोस्फीयर, एटमॉस्फेयर, लिथोस्फीयर, हाइड्रोस्फीयर, ये सभी हमारे चारों तरफ मौजूद होते हैं. इसके बाद में है बायोस्फीयर. द बायोस्फीयर इज मेड अप ऑफ द पार्ट्स ऑफ अर्थ वेयर लाइफ एग्जिस्ट. एंड लास्ट है क्रायोस्फीयर. द क्रायोस्फीयर रिफर्स टू द रीजन ऑफ ऑन अर्थ वेयर वाटर फ्रीजिंग इनटू स्नो और आइस. यहाँ पे ये जो है हमारा जो भी वाटर होता है वो आइस के अंदर कन्वर्ट हो जाता है क्रायोस्फीयर के अंदर. क्रायोस्फीयर बेसिकली हमारा वही पार्ट है जिसमें हम आइस या उसको इंक्लूड करते हैं, स्नो या आइस, यानी कि बहुत कम टेम्परेचर जहाँ होता है हम उसको इंक्लूड करते हैं क्रायोस्फीयर के अंदर. फिर हमारा नेक्स्ट टॉपिक है स्मॉग. तो स्मॉग इज़ द combination of smoke and fog. Smoke is two types: sulfurous smoke and photochemical smoke. So major source of smoke, sulfurous smoke, is industrial and household burning of coal and oil. इसके अंदर जो मेजर सोर्स होते हैं सल्फ्यूरियस स्मोक के वो होते हैं जो भी हमारी कार्बन डाइऑक्साइड जनरेट हो रही है फ्यूल बर्निंग से या इंडस्ट्रीज़ में जो भी हम काम कर रहे हैं, जो भी गैसेस निकल रही हैं, वो इसका मेन सोर्स होती हैं. मेजर इफेक्ट जो होता है सल्फ्यूरियस स्मोक का वो होता है इरिटेशन इन लंग्स एंड थ्रोट्स. और जो पॉल्यूटेंट्स हैं वो मोस्टली होते हैं सल्फ्यूरियस डाइऑक्साइड. इसके अलावा जो मेजर सोर्स है फोटोकेमिकल स्मोक के वो होते हैं एनर्जी पावर प्लांट एंड कोल्ड फायर्ड पावर प्लांट. इसके अलावा मेजर इफेक्ट ऑन ह्यूमन इन क्या होता है फोटोकेमिकल स्मोक का वो है आई इरिटेशन. इसके अंदर बहुत ज़्यादा जब आप स्मोक में जाते हैं तब आई में बहुत ज़्यादा इरिटेशंस होने लग जाती हैं. मेन पॉल्यूटेंट्स हैं नाइट्रोजन डाइऑक्साइड, ओजोन, एल्डिहाइड, पेन एक्सेट्रा. Greenhouse effect. Some of the infrared radiation passes through the atmosphere. Some is absorbed by greenhouse gases and re-emitted in all direction by the atmosphere. The effect of this is to warm Earth's surface and the lower atmosphere. ये ग्रीन हाउस इफेक्ट होता है. अब ग्रीन हाउस इफेक्ट में क्या हो रहा है, जो ग्रीन हाउस इफेक्ट में जो ग्रीन हाउस गैसेज हैं वो बहुत ज़्यादा बढ़ रही हैं पॉल्यूशन की वजह से. इस वजह से ग्रीन हाउस इफेक्ट जो है वो हार्मफुल इफेक्ट दिखा रही है हमारे अर्थ पे, जैसे कि ओजोन लेयर में छेद हो रहा है. इस तरह से जो हमारी अर्थ है वो बहुत ज़्यादा वार्म हो चुकी है ग्रीन हाउस इफेक्ट की वजह से, ग्रीन हाउस गैसेज की वजह से. तो ये एक हार्मफुल डायरेक्शन में जा रही है. फिर है क्लाइमेट चेंज. क्लाइमेट चेंज ब्रिंग्स serious effects. It makes temperature rise, leading to more extreme weathers like storms and heat waves. Ice melt, causing sea level to go up and threatening coastal areas. Animals and plants faces challenges in their habits. Changes. Overall, it is a big problem impacting our planet. Climate change भी एक बहुत बड़ी प्रॉब्लम बन चुकी है हमारी अभी की अर्थ पे. कॉजेज़ एंड इफ़ेक्ट ऑफ क्लाइमेट चेंज. तो कॉजेज़ क्या हैं, जो बहुत ज़्यादा हम पॉल्यूशन कर रहे हैं या जो एनर्जी यूज़ कर रहे हैं और डिफॉरेस्टेशन, लाइव स्टॉक, ट्रांसपोर्ट, इन सब की वजह से जो है क्लाइमेट चेंज में बहुत बड़ा इफ़ेक्ट पड़ रहा है. जैसे कि टेम्परेचर जो है वो रेज़ होता जा रहा है, सी लेवल बढ़ रहे हैं और लैंड डिग्रेडेशन हो रहा है, वाइल्ड लाइफ डिस्टर्ब हो रही है. नेक्स्ट टॉपिक है टॉक्सिकोलॉजी. द स्टडी ऑफ पॉइजन इंक्लूडिंग द सोर्स, इफेक्ट एंड टेम्परेचर, ट्रीटमेंट ऑफ पॉइजनिंग. दिस इज़ नोन एज टॉक्सिकोलॉजी. टॉक्सिकोलॉजी के अंदर हम उन सभी मेथड्स को इंक्लूड करते हैं जो हमें बता सकें या हमें अवेयर कर सकें और हमें जानकारी दें पॉइजन के बारे में या और भी कोई केमिकल्स जो हैं वो हमें किस तरह से नुकसान पहुंचा रहे हैं या पॉइजन कर रहे हैं. टॉक्सिकोलॉजी के 8 टाइप्स हैं: इन्वायरमेंटल टॉक्सिकोलॉजी, रेगुलेटरी टॉक्सिकोलॉजी, इंडस्ट्रियल टॉक्सिकोलॉजी, फॉरेंसिक टॉक्सिकोलॉजी, वेटरनल टॉक्सिकोलॉजी, केमिकल टॉक्सिकोलॉजी, मैकेनिस्टिक टॉक्सिकोलॉजी एंड ऑक्यूपेशनल टॉक्सिकोलॉजी. यहाँ पे हम डिस्कस करने वाले हैं केमिकल टॉक्सिकोलॉजी. तो 10 केमिकल टॉक्सिकोलॉजी जो होती है उसके अंदर हम केमिकल्स के जो केमिकल्स हैं वो कितने टॉक्सिक हैं इस चीज़ की स्टडी करते हैं. और 12 टॉक्सिक केमिकल्स इन वाटर, ये बहुत इम्पॉर्टेंट है हमारे, क्योंकि हम बहुत सारा काम हमारा वाटर पे निर्भर करता है. इस जो 12 टॉक्सिक एलिमेंट केमिकल्स या एलिमेंट्स कह सकते हैं, वो वाटर के जो टॉक्सिक है वो है मीना माटा डिजीज़ मैम। यस सर। सर स्मॉग जो होती है वो एक तो फॉग होती है जो कि हमारे जो सनलाइट है और जो वेपर है उससे जनरेट होती है, पर जो स्मॉग होती है वो स्मॉग जो है वो पहली चीज़ तो फॉग यानी कि सनलाइट एंड वेपर वेज से जनरेट होती है। दूसरी इसके अंदर जो पोल्यूशंस के गैसेस हैं, बहुत सारी पोल्यूटेड गैसेस, उस वजह से जो है स्मॉग जनरेट होती है। हाँ सर, जो ग्रीन हाउस गैसेस होती हैं, जो हमारी लेयर होती है, वहाँ पे कुछ जो अल्ट्रावायलेट रेज हैं वो अब्जॉर्ब हो जाती हैं, और जो सर ओजोन लेयर होती है वो रिफ्लेक्ट भी करती है और एब्जॉर्ब भी कर लेती है, पास कर देती है, ओजोन लेयर से पास भी हो जाती है अल्ट्रावायलेट रेजेस। थैंक यू।
Thank you, Sunidhi. Next is Shreya Sharma.
Namaskar everyone, myself Shreya Sharma. I am pursuing PhD in the Department of Physics, and the topic of my presentation is Soil Health Assessment through Photonic Crystal Imaging for a Sustainable Future. Outline of my presentation consists of introduction, basic introduction, what is photonic crystal imaging, advantages of PCI, future implications, conclusions. So let's start with the basic introduction. So for assessing soil health is a critical factor in evaluating various physical, chemical and biological properties of soil that is for useful in sustainable agriculture, as it involves and influences the productivity and vitality and viability of agriculture systems. Healthy soils provides a supportive environment for plant growth that can lead to sustainable future. The traditional methods of soil health assessments have been crucial in understanding various properties of soils. So soil health is basically the capacity of a soil to function as an ecosystem. Soil processes various that is gas exchange, absorptions, adoptions, oxidations, water and solute movement. Soil properties includes biological, physical and chemical. Some common traditional methods of soil health assessment includes visual inspection, soil sampling and laboratory analysis, pH testing and NPK testing, but they do come with certain limitations. In visual inspection, simple observation of soil color, texture and structures can be made by anyone, but its limitation is it is subjective and can vary among various different observers. In soil sampling and laboratory analysis, we collect soil samples and send them to a laboratory for analysis, but its limitation is it is a time consuming process and may or may not provide a real time data. So what is photonic crystal imaging? So for understanding photonic crystal imaging, first we have to understand what are photonic crystals. So basically photonic crystals are the structures that manipulate and control the flow of light. They can be designed to exhibit several or specific spectral sensitivities, means they can interact differently with different wavelengths of lights. This properties enables us to capture informations about the chemical and physical properties of light. Photonic crystal imaging refers to a technology that incorporates photonic crystal into sensing device or into an imaging system which sense and assess the soil health. The integration of photonic crystals into advanced imaging system facilitates, it provides us a real-time, a three-dimensional visualization of soil structures that surpasses the traditional methods. Advantages of PCI are they provide fast results, they have higher accuracy, they are non-destructive in nature, real-time monitoring, and high spatial resolutions. Future implications include biophotonic crystal imaging. We can have precision farming, we can improve the crop health monitoring, we can optimize the resources that are water and time, we can enhance the decision making. The farmer can enhance his or her decision on the basis of the data provided by photonic crystal imaging. Data driven agriculture can be proceed, and the promotion of sustainable practices. So in conclusion, soil health assessment through PCI offers high spatial resolutions, they have real-time monitoring and precision agriculture that is very useful for agriculture practices that can lead to a sustainable future. They offers various advantages like optimized resource management, early detection of soils issues. They has the potential to significantly enhance the soil health by providing all the advantages that includes real-time, non-destructive insights. They represent a promising future and promising advantage for advancing agriculture practices towards green precision, sustainability, and productivity. These are the references I have used for the review. Thank you. So the fabrication process, we have to incorporate them into sensing devices. That can be tedious sometimes, but it can be done. So I have consulted 5 to 6 papers, basically on soil health and other papers on photonic crystals. Thank you.
Thank you, Shreya. Next one is Ronak, serial number 34. Okay, sir.
First of all, hello everybody, and I thank you so much all of you for being here and listening to me and giving me a valuable time. So my name is Ronak and I am pursuing my MSc Mathematics from Mody University of Science and Technology, and today I have implemented a really interesting thing. One of my favorite teachers has always told me that nature is connected to mathematics and it can always be understood by mathematics. So here all I have done is that I have connected mathematics with environmental sciences using the queuing model. I guarantee you will gain something new in your knowledge after the presentation. And without any further delay, let's dive into the presentation. So this is the abstract of my presentation, which I have. The basic idea behind the presentation is that I have applied queuing model to the restaurant named Vinayak Restaurant in the Sikar city, to see basically how the arrival rate of customers are being there and how the queues are made there of the customers. And I applied MMS queuing model here to see how they can actually improve their overall customer satisfaction, as well as they can reduce their energy consumption through reducing their waiting lines. So this is about the abstract. All the contents are there as shown in the slides. The introduction is as given on the screen. We have observed that there are 60 tables and 10 waiters they are providing to their customers in the restaurants. And I have observed that around 200 customers on weekdays and 490 on the weekend days are arriving in the restaurants. This is the literature review I have used in the research review. The what is basically queuing model? The queuing model consists of two main elements, which is customers and the servers. How the customers are arriving and how the service are being provided to them. The basic characteristic of the queuing system is arrival time distribution, how the people are arriving in the system and how service are being provided to them and how the service channels are working, how queuing discipline are made, like how they are providing the services to the customers arriving there, and the queue length and system capacity. So there can be single server, like single server which is providing services to the customers, and also multiple channels providing services to the customers. So the basic Kendall notation is given here for the queuing model. The Kendall model basically shows how the arrival time distribution is. Here A shows how the arrival time distribution is, and B shows what the service time distribution is, and C shows number of channels, D is capacity of the system, E is the queuing discipline, how the queue is being serviced, and F is the size of calling sources. Here I have used Little's theorem, which says the length of the units in the system is equal to the lambda multiplied by waiting time of the system. So here the basic approach I have used is this. I have applied MMS FCFS infinite model in this paper, which says there are S number of servers in the system and the queue discipline is FCFS. The assumption I have made are shown on the screen. This is the overall approach I have used. The customers are arriving in the system, and there are S number of servers, which I have taken 10 in this model because there are 10 waiters in the restaurant, and then they are getting departs. Like for the restaurant, after getting their dinner or after getting their lunch, they leave the system. So here they depart customers. So the basic methodology that I have used is given on the screen. The formulas I have used: the utilization factor, the rate of arrival of the customer, rate of departure, and rate of service is given. This is the basic formulation that I have used using Little's theorem and Kendall notation, and the basic calculation of the formulas. I have used these formulas. So this is the overall observation I have taken from the restaurant data, and I have seen that on the weekend days, monthly around 1,000 customers are arriving in the restaurant on weekend days, and on weekend days around 2,500 customers are arriving in the restaurant. So I made this graph using the observation. And for the calculation, I have separated calculation for everything, like for service rate, for arrival rate, for length of queue, and all the other parameters. So here I use the formulations and calculated these values. So this is the final calculation where I have seen that the utilization factor is 0.49, and the probability of 0 customers is 0.28. So overall benefit is that we are not only focusing on the enhancement of the customer satisfaction, but we are also focusing how we can resolve the environmental issues using this model. So this is the basic benefit and environmental impact on the from this model on the environment. The less the customers is, the less the waiting line is, the less the energy consumption will be, the less the traffic will be, and hence we can resolve the environment using mathematics basically. And this is the final conclusion that I already discussed. The arrival rate is 1.4, and the service rate is 0.285, and the utilization factor is 0.49. So this is the final conclusion, and the benefit I